Today, we'll be diving into some of the most exciting and rapidly evolving areas, from genomics and transcriptomics to metabolomics, proteomics, and beyond, all combined. We have an incredible lineup of speakers here today and networking opportunities after the session. So thank you all for being here, contributing your time and your expertise. Our next speaker is Dr. Huiwen Zheng.
Thank you very much for having me here. Today, I'm trying to introduce one of the projects we're doing together with Professor Women's lab, that they're generating these novel brain organoid protocols, and we show the power of using multiomics profiling to really categorizing what we have in the system. So to begin with, all right, the biggest challenge, especially in the brain, is its complexity and inaccessible due to its region. And on the left is a figure from a recent study that they sequenced these transcription profile of diverse cell types across the adult brain. And in total, they sequenced about 105 dissections with 3.3 million cells across different regions and also 31 different subclusters in the brain. And this is not only the complexity of the data, and also the biology is hidden in the human, especially in the human brain, that it contains around 86 billion neurons and diverse cell types. And due to the limited availability of the representative data, it is kind of difficult to fully represent the diversity of the cell types in different developmental stages and disease states. Therefore, especially for the computational challenges, even with such rich data of 3.3 million cells, it's very hard and difficult to work with these basic algorithms, or we even need much more memories to make them actually running or load the data itself. That's why we have a great model that tries to simplify but contain most of the biology within this brain organoid model. So brain organoids are these 3D structure models that derive from human and pluripotent stem cells or the embryonic stem cells. Then they were differentiated into different cell types, including neurons, glia cells, including astrocytes and oligodendrocyte precursor cells. And people have shown that this model can have similar profiles of those found in the actual human brain. Therefore, this is a great model that allows us to directly mapping the patient-specific gene expression patterns to disease phenotypes in the control environment. Then we can do a large scale of different drug perturbation and testing and screening, etc. However, there is always, an incomplete model under development, right? Current successfully brain organoid models have successfully incorporated many cell types, especially including neurons and some of the astrocytes, as listed here. But in particular, the related to microglia, oligodendrocytes, and also some of the endothelial and vascular cells is still lacking because it's a system in the dish. It's hard to imagine that you have vasculars and have, oxygen and blood flows into that. Here, especially, we highlighted the microglia cells, which is the brain immune cell that mostly are absent from the standard organoid protocols. So the reason behind that is most of these immune cells are derived from the mesoderm germ layer during gastrulation weeks, and while the neurons are actually derived from the ectoderm. So it's very difficult, kind of rarely you can get these mesoderm cells in the ectoderm-derived brain organoids to have these indigenous microglia gene expression, etc. Therefore, we have come up with this new model, Cerebrus, which is short for CBRs. It's a standalone self-organizing system that endogenously gives rise to all 4 major brain cell types, including different types of neurons, astrocytes, oligodendrocytes, and microglia. And we're able to show some of the gene expressions and gene signatures that can able to recapture the analogs of vascular cells in the data as well. And we use this, we work together with Dr. Gilan from Monash lab doing this project together, where they will show this model can successfully be used in a special case of multiple sclerosis, which is a autoimmune diseases that targeting your brain, basically. To facilitate the discussion today, I've provided basically a study design and overall view of the multiomics data we're actually involved in this project. So we get in-house and public available datasets from single-cell RNA-seq data, bulk RNA-seq, spatial transcriptomics, and proteomics data that has been sequenced or obtained from 4, especially CBRs at 2 different time points to show the maturation, and also a comparison as a benchmarking to the other protocols at the similar maturation rate to just showcase what we can actually provide in this new system. To give you a little bit background, what is different omics I just mentioned? So especially here, talk about the gene expression studies we have in the brain. So thinking about the set of Legos from 20 years ago, we're able to perform bulk RNA sequencing, where we can have an overview of gene expression as a whole, as organisms, as a system. We know what genes has been upregulated or downregulated, and then we are able to pinpoint maybe that's one of the 2 things that is critical to that organisms. But we're lacking of the single cell resolution. So we don't actually know what is the leading contribution from different cell types. Is a particular drug we're going to impact all cell types or rather just one? Then single cell RNA-seq is a really great one to have this distinct gene expression at this as this resolution. But more recently, I think the last 2 to 5 years, we have spatial transcriptomics, where it's an additional add-on that we have this spatial location. So now we're able to not only know what the expression for each cell, and also we know where they're coming from. We can also use this to understand how cell interacts and how cells interact with other organisms that are in the body as well. So to leverage all the omics that have been developed so far, firstly, we just sequenced these CBRs at day 50, which is a very early time point for the initial protocol. So because we're trying to induce both ectodome and mesodome at the same time, and then we just want to know what's the balance and how much we can induce both of the cells in the system. And then we run single-cell RNA-seq data. Although you can see here, the data was pretty consistent, just back shapes. And you can see most of them, each single organize contains all the cell types as basically you need it. You can see most of them, over 50% of the cell type percentage is categorized into mesodome, which means we're no longer having a ball of neurons. We're actually having more of these mesodome cells, which could become muscle cells in the later or something else. Then we use this information to inform our collaborators, like wet lab people, be like, okay, we probably have to optimize the protocol a little bit. We probably have to bring back the neurons to the system where we still can use it as a brain organized system rather than something else. That's why Gilan spent a lot of time trying to optimize the protocols and change the differentiation media time, and then eventually we're able to successfully promote a new CBR or an optimized CBR protocol that on the top you can see that's the immunostaining to show the mature oligodendrocyte, while at the bottom is where you have a immunostainings of global neuronal architecture to prove that this is a good system, that we still have a lot amount of neurons while we have the cell type we really want to see. But as a computational biologist, seeing that is something is not really quantitative for me to actually show that is the cell type we're looking for. That's why we sequenced again with the single-cell RNA-seq, and now we're able to see that the light purple color is the only mesodome cells we have in the data, which is about 5% out of the total cell. That's a good balanced number, that most of them are still related to brain development, about the progenitor cells, neurons, astrocytes, and all the glial cells. That is really critical. On the right is a very typical way of annotating the single-cell RNA-seq data. Normally we use a few or a collection of gene expression markers that we know are specific to one or different cell types, and then that's why we can give a name for them. But given the richness of the data we're having from transcriptomes, a more kind of advanced way that people would do these days is trying to do a projection or label transfer. So here we use the data from human fetal brain atlas, which comprise about 1.6 million of cells, and then we're trying to project our data into it, and we're going to see how similar our neurons is to the actual human neurons and all the other cell types as well. And here we use SCNVI, which is a deep generative model that harmonizes multiple single-cell RNA-seq datasets. It is especially critical for large datasets that you have 1.6 million of cells that it can easily run, and the algorithm can generate a latent representation to do the cell assignment using a probabilistic kind of algorithms behind that. Then you get a probability how much reliable that label is actually being transferred. And we calculate the relative presence in our data by using the aggregate count of the mapped cell identities for each of the cluster from these KN classifications. And you could see that the darker color means the higher relative presence in the primary tissue. Most of our neurons, progenitors, and also the glia cells was highly present or similar to the ones from the primary tissue. But you can see the really light gray on the bottom light, where it is indicating of these newly generated or mesosome or microglia cells. So one of the reasons is also the caveats of single-cell RNA-seq data. The primary tissue doesn't really have a huge coverage of the microglia cells to begin with. And that is also because the technologies and like the fragile of microglia cells were actually a lot of time being kind of missed during the sequencing. But nevertheless, we have proved at least what we know about like the brain organoid itself is truly represents what's going on with the brain, and especially with respect to the neurons itself. The other major issue in brain organoid is cellular stress due to the large hypoxia conditions. As they grow in a dish, they don't have a lot of airflow around that. They're culturing based on media. So studies have found that two major pathways related to ER stress and glycolysis is the main biological pathways that is represent of such cellular stress. Then we compare to another existing method or a organoid kind of protocol published in 2020, where they actually show cellular stress in cortical organoids impairs molecular subtype specification. And we're able to show that by generating this gene signature measurement score, and our CBR is significantly low, shows significantly lower cellular stress as compared to these external organoid protocols. And it's also interesting by time, you actually accumulate more kind of cellular stress as you kind of expected to see. Next, we want to compare to other established brain organoids. So here we use bulk RNA-seq on different iPSC lines that derive into using two different protocols. And we can show on the PCA map, like PC1 basically explains the variance between two different protocols. And the y-axis is actually represents whether these cell lines are coming from patient-derived iPSCs or they are coming from embryonic stem cells. So there is a epigenetic landscape difference where these are regions, but nevertheless, we're still able to show based on these gene markers that CBR holds a high expression of microglia and myelination, similar level of astroglia expression, and also relatively low expression of neuronal cells, because we kind of compromise a subset of neurons to become the proportions in the microglia cells. Then we're also a bit concerned that kind of compromised neurons is going to work in a system we call a brain organoid. We use proteomics to actually sequence and again understand what's like the critical markers that related to like neuronal functions. So here I just show you one example that we found these cortical layer markers that is really specific to the developmental of the cortical layers are actually present or higher expressed in CBRs as compared to the other established cortical organoids, which they have shown they have a higher number of neurons and etc. Similarly, we also show a similarly expression profile of the protein coming from the synaptic markers. And this is where really multiomics jumps into space, that we couldn't find these markers at all at the transcriptome level, but only from the proteomics level. So it really complements the story behind these new model generation. So accepting like zooming from a organoid itself, we're also interested to see a specific transcriptome trajectory. So in the brain, you actually have a set of cells that is called oligodendrocytes. So it kind of initiates from this oligodendrocyte precursor cell, and then it can fully differentiate into myelinating oligodendrocyte that has a function and kind of connections with your neurons. So we use trajectory analysis, which is a computational method to impute or understand the pseudotime of the cells. So we sequence everything at the one time, but we trying to understand what's the transcription landscape, where we can actually predict who comes in first or who comes in later. And then we can draw these kind of like smooth curve to actually infer these trajectory coming from dividing OPC to OPC and all the way to the myelinating OPC. And we're able to show the relative gene expression marker patterns that is consistent with the info lineages, where you have these OPC markers was really highly expressed in OPC only, where these myelinating genes, including MOG and BP and all the stuff, was only exclusively to the later myelinating oligodendrocyte. So that's great. Further beyond that is we also want to compare how other existing protocols have done so far. So to date, there is a integrated transcriptome datasets that leverage 26 different publications, 36 different datasets with 1.8 million of cells. And then they also suggested on the right corner, there is it's an OPC and oligodendrocyte subcluster. So we want to know if the existing protocol can reconstruct the same kind of trajectory analysis, trajectory that we discover with CBR or not. And then we use a canonical correlation analysis, which is more suitable for these smaller data sets, and it can run quicker as well. In conclusion, you could see that the OPC was pretty well aligned with the other established protocol, where you have this little corner with only the myelinating OPCs, myelinating oligodendrocytes highlighted there. And that's also the only one publication so far that have claimed they have myelinating oligodendrocyte from the paper. So that's actually kind of backing up each other. We're able to show these really important cell types happening in the organoid system. Additionally, we also find these really interesting gene called GET1, which is a neurotransmitter that doesn't really, well known to have a function in the OPC or the maturation of OPC. And then we dig into that, and then we figure out that this contribution of OPC in GET1 in OPC maturation is exclusive to the protocols where we have a mature myelination. And around the right side, this dot plot, this is the only protocol that's similar to the left-hand side that claim they have both MBP, which is the myelin protein that helps to make up the myelin. So it's also a new to show the power of this new model that we can recover some new mechanisms that we couldn't do much before. We don't have a mature oligodendrocyte in the brain organoid. Further on, we look at the different subtypes of the microglia that are marked by different, really diverse, different gene signatures that people, use in different protocols. More interestingly, we actually find a more consistent expression, PLXDC2, that marks all the subclusters that are related to the immune responses or a microglia. One major thing in single-cell RNA-seq or transcriptome field is it's really hard to identify or annotate a cell type based on one or two markers because they are most likely inconsistent. They are not as consistent as we see in the immunostainings or the experiments. Sometimes you have to use a combination of different panels because of the sequencing issues, because of the the data representations or just that snapshot that genes that don't express at all. So we want to propose a PRX-DC2 as a novel, robust microglial transcriptome signatures that can really conserve that shows conservative and across different sequencing platforms. We use these integrated data sets specifically for the microglia atlas, including 241 samples and 91,000 nucleus and cells. And could see that compared to other two canonical marker, which is alpha-1 and TM119, this PRX-DC2 shows a consistent expression in over 94% of the microglia across different age, different disease types, and different phenotypes as well. More importantly, we use spatial transcriptome to actually understand what's the spatial location of this particular gene in the developmental brain. So we leverage this recent published Cell paper where they sequenced this brain, like actual fetal brain, across different gastrulation weeks. I could see that compared to the other two canonical marker, this one is another one because we can't find alpha-1 in these data sets, as I just mentioned before. PRX2 actually can mark all these very clear trajectory and locations as compared to the other ones that is more sparse, and showing its ability that can really use in a multi-platform and multi-scenario cases. As I mentioned, we're really trying to establish these models to understand different diseases. So here, we're trying to understand multiple sclerosis, which is a chronic autoimmune disease where immune cell systems attacks the protective myelin sheet of nerve fibers in the central nerve systems, disrupting nerve signals and causing a wide range of neurological symptoms. So the two hallmarks of MS is one, definitely demyelination, the loss of axons, and then two is the inflammation. And this is something we couldn't really model from the brain organoid because we don't have these two cell types that represent what we need for this particular disease. So to induce MS, we use laser testing. And then you can see that once we put the laser testing for 48 hours, and then we wash that away, and then let it recover for another 48 hours. You can see the loss of myelin and loss of myelin here with the GFP, and also a reduction at the neuronal function as well. Using bulk RNA-seq, we're able to identify a pattern here. Where you find these sort of genes that upregulated at T1, which is 48 hours of treatment, and kind of recover a little bit after you wash it away. When we look at the particular functions related to these genes, we find genes related to inflammatory responses and cytokine-mediated signal responses as well. Here I just show a few of the markers that is related to different chemokines and interleukins. We're also able to show these amazing loss together with microglia and SOX10, which is the OPC, kind of mimicking what the microglia or macrophages is eating, the destroying myelin, that just reflecting what's happening in the brain of a MS patient. Lastly, since we have this brain organoid bulk RNA-seq data, we thought we want to estimate how similar they are close to the patient data. So we use this data, bulk RNA-seq atlas coming from these brain macrophages transcriptome data in these MS patients that published a few years ago. And then where we project our data, which is in the triangle, to the PCA space of the entire patients that is either a control, a subtype of MS, or a more serious subtype of MS. And we could see that at different time points, for instance, these control at T0, the pink ones are more close to the control in the MS patients, where the treated ones are more likely to be the MS patients. And then you can see here from the heat map as well, this is the control in the CBR, which lies within the healthy control of MS patients. And then similarly, this is more likely to be like the ones that has been treated. That is really great to see that even if brain organoid is still a simplified version of what's going on with your actual brain system, we're still able to see those transcriptional similarities and also the real power of using these new models to understand different diseases. Therefore, in conclusion, we have presented a new method for generating human brain organoids, a 3D system that formed by all cell types present in the brain. And CBRs promotes the presence of multiple mature cell types, creating a cellular environment that closely resembles of the human brain. And leveraging the multiomics approach, these comprehensive molecular signature with key pathways that mimicking the primary brain functions can be reviewed. And also the usage of computational methods such as label transfer and probabilistic variational inference methods can support the utilization of these multiomics data and also benchmark with atlas-sized single-cell RNA-seq data and resources. So with that, I'd like to acknowledge the group members from my lab and also our lovely collaborators from the Women Time Lab to generating and then co-design all these amazing experiments together. And also for Novogene's input to give us really quick response and also help with generating these highly reliable transcriptome data sets. So thank you all for listening, and I can take some questions. Yes?
Okay, do we have any questions in person? Yeah.
Yeah, really cool talk, very clear. I have been very impressed by bioinformatic analysis. I have to say I'm really interested in the biology of the organoids.
Yeah.
What did you do differently from previous approaches? Sorry. No worries. Yeah, what did you do different to previous approaches that have allowed you to grow these more representative organoid systems?
Yeah, so I can't tell you exact details and medium and everything because I don't step in the lab.
Okay.
But what I can say is previously a lot of protocols are really pushing to have different neuron subtypes. So they're really pushing towards the ectoderm. And here we trying to go back a little bit. We use the undirected method. So instead of just pushing into one, we give these different growth factors and the medium and then different induction time to push different trajectories of the different gem layers. Yeah, if you're interested, I'm happy to connect you with my collaborator, and then you probably can talk more. They probably can talk more about how they do differently.
Sure. That was great. Thank you.
Thank you.
Hi, thanks. That was a really good talk. I was just wondering if you know how the SC Envy, that you use there, how is that better or different than just, I hadn't used it before. I was just wondering if you know how that's better than just say overlaying like the signature from one data set, like from cells from one data set onto the other. Like what's the benefit of using the SCNV?
So computationally, there's a lot of methods you can do this. And one thing we really care about is time efficiency. And like some of the method will run for days and then you couldn't make it run with your local computer. And also because it's a probabilistic environment, so they give you that kind of buffer, where exactly is more likely to be one label or not. Some other label transfer will just tell you, okay, this is more, together with, for instance, neurons, but you don't know how much reliable that is, right? It could be just out of 1,000 signatures, there are just 2, but they don't have anywhere to put the label, so they just put it as a neuron. So that's the power of the probabilistics models.
I'd be interested to know if budget allows, do we have plans of integrate more layers of multiomics, like for example, metabolomics and
yeah, for sure. So if budget permits, of course, we're really curious, like firstly, we really want to do a spatial transcriptomes on these CBRs because since it's a more undirected protocols, we're curious to see what kind of structures that can recapitulate, right? We're not pushing to one area of the brain, so we're really curious to see what's going on within the system. And also for the metabolisms, of course, we also, especially for modeling diseases like MS, we're also curious like what's going on once you treat it. And we have some potential drugs that we want to screen as well, and we'd like to see how those responses to that differently.
So as a bioinformatician, because you incorporate so many different omic layers in your presentation, and that was really beautiful, by the way, what do you think is the challenge that you encountered to try to analyze all of these data?
That's a very great question. I think the challenges actually comes from a deep understanding of the biology. So for instance, you provide services that they already can do, mapping and the Ganda-Camp matrix, and you're able to start understand what's going on, what gene has been overexpressed and underexpressed. But the most challenging thing is how can you tease out that signal that is relevant to your biological questions? What is like the more advanced method that can help you better understand what's going on, and how can you incorporate with these well-acknowledged and well-established atlas-sized data that people use these days. They will ask you like, what about this? What about that? Like, how can you really make consistent conclusions coming from different data sets? Like, at least that's something really challenging for me. And a lot of data sets was not really in a really good quality state as well. That's also extra challenges. We're like, how are we going to do QC? How can we make sure we can include them? That's a really hard decision for us to make as well if the data was not great enough to begin with.
Thank you very much.
Thank you.
Right. Thanks for the opportunity to speak with you today, and to those people online, thanks very much for coming along. I think one thing that's evident at the moment is that the title I gave for my talk is different from the title slide that I've used in the presentation. That it's a variation on a theme, and I think that you'll appreciate where I'm going with this, in the sense that in the microbial world, at least, I think we're starting to move beyond simply DNA sequencing and building a picture based on computational information, and we recognise that there's a continuing need to expand bringing genomes to life, with the view that multiomics approaches can extract more information and meaning and translation into opportunities and new strategies to deal with environmental health as well as medicine. Let's see, how do I advance the slides? Oh, I can just click on the mouse button here. Okay, there's my disclosures. I think I'm going to have to stand still, which is a little different to me, and I'm still showing someone else's background. Oh, okay, all right. So, yeah, my disclosures. And really the microbial world, or the study of it in relation to health and disease, has gone through some evolutionary steps, some of them quite drastic over the last 20 odd years or so. They've been driven by the original omics-based technologies, principally DNA sequencing. And what it's done is it's kind of broadened the appreciation and awareness and the need to consider that health and disease aren't necessarily germ theory of microbiology, where you think of one organism at a time or a pathogen, and through Koch's postulates you study pathogenesis and virulence factors, and the only good bug is a dead bug sort of approach to health and wellness. And more so through sequencing and omics-based approaches at a DNA level, a recognition and understanding that what might look as random chance in terms of the types and kinds of microbes that you have present, there are associations at an ecological or a community-based level between the composition of the microbiota at different body sites and its impacts not just on health but disrupting homeostasis and as a consequence being associated with disease functions. So we've emerged into a future whereby we're starting to think more about ecological principles and the community as a whole, not just in terms of promoting health, but how changes within that community, both in terms of the types and kinds of microbes that are there, but also the expression of their genes and their genetic information influences those sorts of situations. So to think about the ecology of the gastrointestinal tract in relation to the microbial world, biogeography is one way of you doing it and thinking and using principles in macroecology to consider it. So historically, biogeography has been the study of geographical patterns and distribution of species and how those species adapt to those different environments. So we don't find polar bears, for instance, in the middle of Australia, and likewise we don't find the sorts of marsupials we have in Australia in other continents. Even though the climate might be suitable, the predatory features associated with those environments and Australia's isolation has played a key role in the evolution of the biogeography we have in this country. So if you apply those principles to the microbial world and along the gastrointestinal tract, they start to make sense. So if you think about the various features that we have in terms of the chemistries and environmental fluctuations from the stomach through to the large intestine and colon, we can see that there are variations in a number of different parameters, oxygen being a key one. So organisms that have a capacity to use at least limited amounts of oxygen or tolerate or even thrive with it are located in regions where we might find that the atmosphere has a higher tendency to provide that sort of molecule as a source of substrate. But as we move further down into the gastrointestinal tract, and those organisms essentially consume the oxygen faster than the rate at which it can be replaced, we start to shift to a different type and variety of microorganisms, many of them not only being anaerobic in nature in terms of how they grow and thrive, but actually being intolerant to exposure to that particular molecule. And the sacculation and the surface area that's associated, especially with the large intestine and the colon of individuals, serves as a rich reservoir to maintain a very dense and diverse population of microorganisms. And so the point I'm trying to make is if you think of the hollow or the luminal contents throughout the gastrointestinal tract, microorganisms are detectable, but the types and kinds of microorganisms that you potentially will find are a reflection of the physicochemical conditions that exist there. And they vary as we move from the top to the bottom of the gastrointestinal tract. Now, using micrographic-based approaches, it was recognised that at least on the surfaces of the intestinal wall, that there was a presence of microbiota. And what I'm trying to show with this slide is that in addition to luminal contents and stool being the most widely accessed, readily collectible and analysed sort of material associated with the human body, there is a discrete and separate population that specialises in its adhesion and interaction with the mucosa at different gut segments. And not only can the types and kinds of microorganisms within that specific community on the mucosal surface differ throughout different gut segments, so too can the load or the density of that population, its formation and maintenance of a biofilm, and the complexity and the overall size of that can be relevant and important. And so while this has been recognised for many years in a visual context, the sampling, characterisation and the quantification of this population really has been quite elusive. And in addition to that, with the advent of the omics-based technologies and the recognition that the microbiota are at least associated with maintaining health or triggering disease, there is a heightened interest in these mucosa-associated microbial populations and how they may relate to those particular conditions. If I use something like Crohn's disease or ulcerative colitis as examples, these are diseases of tissue. The person gets ill because of dysregulated immune response and inflammation of tissue. And the microorganisms that are in proximity to that tissue may be the key drivers and responders in terms of healing as well as triggering the disease. So unless you're studying that population, or at least sampling and starting to characterise it, it's difficult to understand the full impact of the microbiota in the context of human health and disease. So here in Brisbane, over a number of years, this is kind of a retrospective talk, but in some respects, we've tried to take innovations in a variety of different ways which have been advanced and scaled up by a range of activities that are happening here at the Translational Research Institute now. But our specific niche, literally and figuratively in this space, has been to look at the mucosa-associated microbiota. And the innovations that I refer to as the Brisbane approach relate to sampling and trying to quantify the biofilms resident on mucosal tissue at different gut segments, and also try to bring the genomic information that we've derived from tissue samples and the microbiota that's associated with that tissue to life in the form of mixed consortia that are holistic and representative of the microbial communities at that time of sampling, and offer us new opportunities and advantages in terms of trying to not only characterise those populations, but start to use other multiomics approaches, as people refer to them now as, in terms of gaining insight into the biology and not just the genome of these particular populations. So really the first innovation had nothing to do with DNA sequencing or what might be referred to as omics-based approaches. It was this concept of Professor Holtman, who's the Director of Gastroenterology here at Princess Alexandra Hospital, of being able to collect tissue at different gut segments in a way where you may be able to protect that tissue from exposure or contamination to other luminal contents during an endoscopic procedure. And so he developed this patented device which basically takes standard forceps and puts them inside a sheath, okay? And that sheath is sterile, it's plugged, and so the forceps are not exposed to luminal contents or saliva during the endoscopic procedures, and they're really only extruded outside of the sheath at the time that the tissue sample is being collected. Then the tissue and the forceps themselves loaded with the tissue can be retracted back into the sheath, and the working channel of the endoscope is typically purged with nitrogen gas, so it's oxygen free. And so what this provides a person like me as a microbiologist is an opportunity, once tissue is collected in this means, it's truly aseptically collected and not contaminated with microbes from other parts of the body. But it also allows us to rapidly and effectively store and preserve that tissue under anaerobic conditions, and at a later date, through cryopreservation, bring that tissue back to life in terms of the microbial world that's still attached and associated with it. So in addition to being able to just do standard DNA extractions and use various DNA sequencing technologies to identify the genetic component of the microbial world on those tissues, we can do things like a quantitative PCR-based approach, which is more specific and not influenced or impacted by contamination from external sources. But importantly, for someone like me who's been trained as a microbiologist for the last 4 decades, I can actually grow these bugs in culture under a laboratory setting, often for the first time. And so I'll give you some examples in terms of how that innovation in the clinic has helped to advance some of the work that I'm interested in doing. So we've collected biopsy tissue from the duodenum of various subjects that have been coming in to the endoscopy suites here at PA Hospital for precautionary assessment for either gastrointestinal symptoms, to identify whether there's a real organic reason or pathology that's driving their symptoms, and likewise persons that are coming in for a routine or a surveillance sort of assessment of the gastrointestinal tract in a preventative way in terms of disease. And so basically what we have access to as a part of that is persons that are diagnosed with what might be called a disorder of gut-brain interaction, in this specific case functional dyspepsia, which tends to affect the upper part of the GI with painful symptoms. And then we have a group of persons that we call controls. Insofar as they don't report gut symptoms, we find no organic or pathological reason for any abnormality within the tissue during the endoscopic procedure. And as a result, we feel that these persons are as close as what we can get to a non-DGBI or a healthy sort of individual for comparison. So we use the biopsy forceps as part of that endoscopic procedure to collect that tissue. And then one tissue sample is extracted for total DNA recovery and PCR-based 16S profiling of the communities. And the second one is used to facilitate the preservation of the microbial world in a glycerol-containing cryopreservative. And so some of the features that we've done by using qPCR is that in addition to the standard 16S microbiota profiling, identifying that there are differences in the community shape and structure between persons deemed to be healthy compared to those with a disorder of gut-brain interaction, we've actually been able to monitor in a subgroup of the patients the load or the size of the biofilm that's present on their duodenal tissue. And that what is shown here on the bottom of the figure is that in relation to the bacterial load that we measure on tissue from these subjects, persons that have a bigger biofilm on their duodenum based on these assays experience increases in gut symptoms during a nutrient challenge test. So they drink a fixed volume of liquid within a particular period of time, and depending upon the way they're feeling in terms of bloating or pain associated with drinking that liquid supplement, they will report how they're feeling. And so what we found is that when persons report a more severe set of symptoms after that nutrient challenge test, they tend to have a higher bacterial load on their duodenal tissue. Likewise, if we ask them to fill out a questionnaire as part of their clinical workup before endoscopy and outline how they feel when they eat food or drink drinks during meal times, what we've also found is that their quality of life scores, how they say they feel after they've been consuming food or having something to drink, those numbers drop as the microbial load on tissue actually increases. So in addition to there being particular types and kinds or a community of microorganisms associated with a disorder of gut-brain interaction, it appears that the severity of the symptoms that those persons might experience relates also to the size of that population that's present on tissue. We've also used these sorts of techniques to, as I said before, preserve the tissue and then cultured the duodenal microbiota in this instance from the tissue samples. And using that community and shotgun metagenomic sequencing and other tools and techniques, we've been able to start to produce deep functional understanding of the microbial community through shotgun metagenomic sequencing. Now, that's a technique that's widely applied with samples with a high microbial biomass, such as stool, and sequencing depth can be important in terms of providing you with the depth and the breadth of information that lets you know what the functional attributes of that community is. By undertaking culture and producing that microbial biomass in much greater density, we can apply that technology to start to see what functional attributes are inherent to the communities and are there differences with these particular conditions. And this is work done as part of Lauren Schuther's PhD program, and there were a couple of things that came out of this particular work. One is that depending on symptom severity, so persons that experienced upper GI symptoms referred here in blue as FD or functional dyspepsia, with persons that have more severe and extensive symptoms, which includes upper and lower GI symptoms, which we call functional dyspepsia with irritable bowel syndrome overlap, FD-IBS, we can see that there are distinct differences in those microbial communities, even at the duodenum, that are represented by the metagenomic information that we've recovered from those organisms, from those persons. The other point I want to make is that we've been able to culture a number of these microorganisms and start to characterise their immunogenic potential, let's refer to it as that way, that might be associated with these particular conditions. And I want to draw your attention to this particular strain of Streptococcus salivarius that we've isolated from a person suffering with a disorder of gut-brain interaction. And what we've been able to show in subsequent studies is that surface features of this bacterium specifically, so not other forms of Streptococcus, but this one in particular, when we use serum from persons diagnosed with a disorder of gut-brain interaction, we find a high level of seropositivity present in their serum towards antigenic components associated with this bacterium, Agira3. Whereas in persons without a diagnosed DGBI or not reporting gastrointestinal symptoms, they're almost entirely seronegative towards this bacterium. So we've generated a patent in relation to this differential response in humoral immunity towards a specific duodenal bacterium as a possibility to develop new serodiagnostic tests rather than going through expensive and invasive processes like upper GI endoscopy to identify whether they've got a disorder of gut-brain interaction or not. I also want to talk about some recent work we've done in collaboration here with QUT and the Centre for Microbiome Research, where we've taken consecutively recruited or samples from consecutively recruited patients through the endoscopy suites at PA Hospital, and we've had an opportunity to collect tissue samples at various gut segments and then undertake an assessment using the same sorts of approaches I've just talked about to profile the microbial communities at different gut segments with people classified or categorised to suffer from different conditions, but also add a quantitative assessment to these populations through the qPCR-based approaches I just talked about. We published this in Frontiers in Microbiology late last year. So we wanted to assess and compare the mucosa-associated microbiota at upper and lower GI segments. Our upper gut segments are the esophagus, the gastric antrum or the stomach and the duodenum, and the lower GI segments are the terminal ileum, right colon and rectal biopsy samples. We had biopsies from consented patients. 59 of them were deemed to be controls. So these are persons negative for colonic disease, negative for esophageal gastro diseases, and also symptom negative in terms of reporting gut symptoms. We had samples from ulcerative colitis subjects as well as Crohn's disease subjects. And we've examined the microbial community profiles we see using the different techniques that we have in the context of health and disease, use of proton pump inhibitors, which is really one of the most widely used medical interventions in human health today and has a major impact upon the bridge or the barrier that exists in terms of acid barrier blocking progression of microbiota from the upper GI to the lower GI. We've used something called the SAGES score, which is a structured assessment of gastrointestinal symptoms. So everyone's asked to fill out a questionnaire in terms of how they feel in relation to those sorts of GI symptoms. And a subset of subjects also did the nutrient challenge test as part of their workup for the endoscopy procedures. So what have we found? Okay, so the quadrants really are self-evident, but you can see the top half of these figures represents the upper GI and the bottom half of the heat maps represents the lower GI, and the relative distribution of microbes and their abundances is a mirror image of each other really between upper gut segments and lower gut segments. So our IBD subjects that show their relative and absolute depletion of specific groups of bacteria was also identified through these mucosal tissue samples. All of this work is done with biopsy tissue collected at different gut segments. And that's consistent with the so-called microbial dysbiosis that we typically see in persons suffering from inflammatory bowel disease. So the data suggests that we've got a sound set of data to work with and to compare, and because we've measured microbial load on tissue as part of this work in parallel, we can actually start to talk about an absolute abundance of microorganisms on tissue as compared to just a compositional assessment. So when we look at some of the data and those that showed statistical significance, if we look at the mucosa-associated microbiota in terms of its absolute abundance on tissue at the ileocolonic region, just as we shift from the small intestine to the large intestine, just proximal to the appendix, basically. If you look at the microbial community we see on tissue there, we see that there is a clear separation that we find in the absolute abundances and profiles of the communities between those that don't take PPI, shown here in blue, and those who do. Likewise here in terms of their assessment of their gut symptoms, persons with high gut symptoms scores are separable and distinct to those that report a low SAGES score. And in the nutrient challenge test, which I tried to describe before, which is when you take that volume of drink and for a period of about 30 to 45 minutes report how you feel, those persons that reported the most severe sorts of pain or distress following their drink do tend to separate relative to those that report no or a low sort of symptom response in relation to that test. So again, what I'm trying to say to you here is that it's not just the types of kinds of microbes that might be present, but there does appear to be an influence of the absolute size of that microbial population on tissue that can be linked with these different outcomes. So again, I just go back because I'm changing tack. I've really talked so far about our application of technologies as it might relate to the upper GI, and now I'm going to talk about the large intestine in the context of Crohn's disease. And so what? Who cares about the mucosa-associated microbiota in Crohn's disease? This figure helps tell the story. So this paper is one of the few that's been published that looks at children suffering or newly diagnosed with Crohn's disease. So these persons are also treatment naive, which is what makes them inherently valuable in terms of the study population. So I'm not saying that the changes in the microbiota cause the disease, but it's not been influenced by any sort of medical intervention at this point in time. It's treatment naive. And what did they find when they looked at mucosal tissue samples relative to stool? The first thing is that they started to help facilitate the establishment of this concept of microbial dysbiosis, whereby we see select bacteria shown here in red that are enriched in persons with Crohn's disease, new onset Crohn's disease, and these ones down here in the bottom in green being in lesser abundance or depleted in the same individuals. And when we start to examine the abundances of those organisms on tissue relative to clinical assessments of the severity of the Crohn's disease in these children, we see that there was a pretty strong positive correlation between the abundances of these organisms here in red and severity. So more of these in red, the more severe the symptoms were clinically considered to be. And likewise, reductions in the abundance of these bacteria shown here in green also coincided with more severe clinical assessment of disease, suggesting that there's a yin and yang sort of situation that's taking place here. And obviously, the Holy Grail is finding ways to counteract the proliferation of the organisms in red while preserving the network of interrelationships of the bacteria in green, with the view of either thwarting the onset or driving the remission of persons that have had active disease. Now, where the colonic or the mucosa-associated microbiota fit within this context is that the patterns of microbiota on tissue were much better representative of what these trends were shown to be than what stool samples were. And it's really, there's only one or two publications of this particular type. And so why is it important? What it's suggesting is that the stool sample ultimately takes on an appearance of dysbiosis, but at that point in time, the disease is relatively advanced and it's diagnosable. Whereas the mucosa-associated microbiota, which have been inherently less studied in the context of inflammatory bowel diseases, show these patterns early. So the idea in the context is by better understanding the mucosa-associated microbiota in the large intestine, do we have a more forewarning of disease onset than what we might get through working from stool. And so that's been a key focus of our projects for a number of years now, and has been supported in large part by funding from the Helmsley Charitable Trust for something called the Enigma Project. Now, in this project, I won't go into the details, but the rationale was that Crohn's disease has often been thought of, especially through the 20th century, as a disease of Western countries and European ethnicity, because that's where it was most frequently apparent. In Southeast Asia and the subcontinent, Crohn's disease throughout the 20th century was rare. And it was deemed that part of the reason it was rare was that the genetic susceptibility did not exist in those persons. But subsequently, as we moved into the 21st century, and the socioeconomic status of these regions has continued to rise and improve, two things have happened. One is that those countries have become more westernised in terms of their environment and lifestyle. They live more built environments. Okay, antibiotics have been introduced and are more widespread and more widely used. Food has changed. There is still traditional cuisine based on ethnicity, but processed foods and the introduction of Western foods into the diet has accelerated in these countries over the last 30 years. So our study was to examine a population of persons in Australia where Crohn's disease is considered to be of one of the highest incidence rates anywhere in the world. And with high prevalence, there's about almost 200,000 people in Australia now that suffer from Crohn's disease. And compare that, in our case, to persons from Hong Kong, where we're seeing a low prevalence of Crohn's disease still, but its incidence rate is rapidly rising. And if you think of Hong Kong from a social and economic context, it's a real hybrid between Western and Asian communities and lifestyles. And we've also got additional parallel work done with samples from mainland China. So what we've tried to do in this particular study is work across a gradient of populations from Australia through Hong Kong and into mainland China, and then with support through various charitable foundations, start to ask how similar are the microbial changes that we're seeing in these populations in the context of Crohn's disease in a country like Australia, where we've had it for a long time, as compared to Hong Kong, where it's more emergent and rapidly rising, and then in mainland China, where it really is also really an emerging disease, but because of the population size and the incidence rate, much more people are suffering from it. So that's the context of the Crohn's disease or the Enigma project. But basically what we were challenged to do is identify a way whereby we might bring the microbial genomes to life from the mucosa-associated microbiota. And I guess this is starting to lead into what I'm referring to as looking through the looking glass approaches using multiomics techniques. So again, like I've referred to a little bit in terms of the duodenal microbiota studies, what we've done in this particular work is biopsies have been collected and have been rapidly stored in a cryopreservative to protect the microbial community from exposure to oxygen, and then subsequently culture those microorganisms using a habitat simulating media that facilitates us producing what we believe to be a holistic representative culture of the microorganisms recovered from the tissue samples of those persons, and then use various omics-based technologies to start to characterise their functional attributes and potential. So we do that computationally, but I'll also start to refer to some more functional assessments as well. All right, so what? Who cares? This is giving us a measure of tools that may be country-specific in terms of diagnostic potential for whether a person might be monitored and deemed to be healthy, or their population starting to change, predictive of Crohn's disease. Here we've started to use the microbial cultures in and of themselves to start to characterise how, at least in a laboratory setting, those populations may shift or change in response to their exposure to different nutrients or food groups that are typically encountered in persons that suffer from these conditions. And so what I'm trying to show you here are in vitro growth of microbial cultures recovered from tissue of Crohn's disease subjects post-operatively. So these persons have had surgery as their treatment strategy for removing their Crohn's disease and to essentially take all the inflamed tissue out of the colon. And then 6 months later, we've been able to recover biopsies from these persons on colonoscopy, and we've produced microbial cultures from these individuals. And then we challenge them in the laboratory to a medium that has no additives or contains small concentrations of two of the widely used food additives in processed and ultra-processed foods, those being carboxymethyl cellulose or polysorbate 80. And then we have the third treatment arm where we combine both the food additives together. And principally what I hope you might be able to recognise here is that each colour represents the microbial community from a different subject, and the more closer those symbols of the same colour are, the more similar the populations are to each other. Okay? So for 4 out of these 5 persons, the addition of either one of these food additives or combination really didn't have much of an impact on their microbial community. It was only when we started to use a particular microbial measure afforded by the DNA sequencing technologies, where we could not only just assess the types and kinds, but the relative abundance of these microorganisms, we started to find that one subject, in response to these 2 food additives, either alone in combination, had a big shift from the so-called control to all of those being greatly different. And the differences related to these 2 bacterial taxa, Proteus and Veillonella. And these microorganisms, in other words, their population and the size that's present in that community is enhanced by their exposure to these two food additives. And it was only this one person, CD34, that had these two bacteria within their microbial community. So again, what I'm trying to say here is it gave us an opportunity to start to link food additive exposure to specific microbial taxa that we've been able to show by other assessments have various drivers of Crohn's disease pathogenesis. They're pro-inflammatory, they're in high abundance with active disease, they're associated with the gut mucosa, just like these ones were recovered here. And so basically studies like this, it would be really hard to kind of make a prediction from just DNA sequence data alone that these food additives would have this kind of personalised and stimulatory effect towards two key bacteria considered important to Crohn's disease pathogenesis. And so by bringing the genomes to life and having the microbes in culture has allowed us to start to predict who may be responsive to these food additives and who might not be. And importantly, provide recommendations to those individuals which foods they should, or food additives are okay for them to have a little bit of, but ones that they should avoid to minimise the impacts on their populations and the potential to trigger their disease. By having the microbial cultures, we're also able to start to look at the small molecule production or the metabolome or proteome that arises from these bacteria. And this is work done by Zheng, who's basically helped us show that 3 of the samples or secretomes from 3 of the subjects were deemed immunosuppressive, whereas 2 were from persons and those secretomes were immunostimulatory. And then we can go back and basically identify that the microbial communities in our 3 persons immunosuppressive are clearly different and separable from those that are immunostimulatory. And then finally take that to the next step to start allowing us to identify who are the discriminatory bacterial taxa that appear to be driving an immunosuppressive effect and the ones that are driving an immunostimulatory effect. And again, just that link to Veillonella and to Proteus via Morganella being strongly associated with communities that have a pro-inflammatory output that interacts, at least in an in vitro assay, with mammalian epithelial cells to drive IL-8 overproduction and inflammation. The third thing that we've done once we bring the genomes to life is that, again, we can harvest these secretomes and in a moderate throughput assay, make an assessment of their immunomodulatory potential with in vitro mammalian epithelial cell lines and start to identify which individual bacterial species may be responsible for producing bioactives that have an immunosuppressive effect. And this work is carried on within the larger context of TRI involving persons from Marta Research Institute as well as Institute of Molecular Biosciences. And we've afforded the isolation or identification of the bacteria, and our colleagues in the clinic as well as at IMB have looked at the structural features of the bioactive molecules with the concept of developing a drugs from bug kind of approach to new therapeutic interventions that suppress inflammation in the context of inflammatory bowel diseases. So basically what I've tried to share with you today is not so much highlighting the different omics platforms or the techniques or instruments that we use, but why they are important to have access to and are available, and what it is we need to do to capture the biology so that we can use them most effectively. And for us at least, with our interest in the mucosa-associated microbiota, it allows us to better navigate the microbial landscape. It's bringing microbial genomes to life, and it allows us not just to do a functional characterisation of these communities, but actually be a bit more hypothesis driven in terms of what we're looking at. Okay, so it gives us a chance to define the metabolomic and bioactive landscapes, not just of the consortia themselves, but of individual species that we may isolate or recover from these microbial communities. It allows us to look at what I call chemical ecology in health and inflammation, as well as active disease and also remission. Okay? It's taking us beyond just trying to categorise things by an inferred function using reference databases like KEGG or KEGG ontology groups to actually start to get the biological outputs and the chemistries that can be analysed and characterised now using the sorts of technologies that we have in multiomics. And it enables and translates into what I call a predictive ecology. So if you go back to the original sentiment I was sharing with you, where disease involving microbes was considered largely to be infectious disease, and if you follow Koch's postulates, you could correct that condition and cure persons, to now starting to think about how, as a community, we can modulate or shape it so that that pattern and community as a whole maintains health and is resilient to the alterations referred to as dysbiosis that disrupt gut homeostasis and drive inflammatory responses. And I think the work that we've done with food additives in the microbial cultures is allowing us to identify biomarkers within the microbial populations and help us guide dietary advice so that we help people avoid triggers, particular food additives, and also promote the onset and maintenance of good bacteria as a community as opposed to one organism at a time. So I guess the take-home point and the final message for me this afternoon is that DNA sequencing has been fantastic. I mean, it's been a great step advance, and I've been fortunate enough in my career to take advantage and gain new opportunities because of it. But as a microbiologist, I'm thinking we're entering into an era, and I think TRI, through the Centre for Microbiome Research, as well as other groups here in Brisbane, recognise the role of biology and microbiology. It's not just microbiome, it's microbiology, or microbial biology, even more specifically. And so what does that mean in relation to multiomics? It means it's going to give us the opportunity to use that instrumentation and approach to look at what microorganisms do and produce, and work our way back into the genetic information to facilitate identifying the regulatory and the ecological features that influence the outputs, and as a result, link that hopefully in a more personalised way with managing disease and health in various persons that suffer from conditions like inflammatory bowel diseases. I'll wrap it up there. Hopefully I haven't gone too long, but just to thank everyone. Some of these people are in the audience, so thank you for coming along. But my colleagues at various institutions in relation to the Enigma project, and more locally here at QUT and at the PA Hospital and UQ. Thank you very much.
For the lovely talk. I'm glad you still bring out the Brisbane approach, so I did mention something correctly.
Yeah. You did exactly what you had on the piece of paper.
Exactly. Do we have any questions from the crowd? Okay.
Really fascinating talk. Thank you very much. I was just curious, is there any evidence for a link between gut microbiome and response to COVID and long COVID in particular? I know long COVID symptoms often, one of the main symptoms is often sort of gastric symptoms, and I'm just curious if you comment on that.
Yeah, very quickly, my colleagues at Chinese University of Hong Kong have been afforded a real opportunity that's globally unique to study this. And just in a nutshell, gastrointestinal symptoms and insomnia in particular are major kind of signs or symptoms of long COVID. The gut microbiota, looking at the stool, is highly associated in terms of its features with those elements. Fecal microbiota transplantation has been shown to be efficacious. A multi-strain form of probiotic comprised of bifidobacteria species developed in Hong Kong has also been demonstrated to reset or restore a microbiota in long COVID patients that is persistent and reduces the symptoms, especially of insomnia, but some of the other gastrointestinal complaints that people experience. So, short answer is there are relationships that have been identified between stool microbiota and symptoms in long COVID patients, and the placebo-controlled trials in Hong Kong with their probiotic formulation, and more recently with fecal microbiota transplantation, shows that by resetting the gut microbiota in the colon, you can bring about improvement in symptoms and alleviating some of the bad news aspects of long COVID.
Thank you.
Thank you so much for your talk. It's really fun to see on the other side from human diseases. So I'm just wondering, in your vision particularly, you mentioned about using these microbiomes as a predictive markers and the advisement for what to avoid for a particular patient. What is your vision to bring in human cells, like the interactions from the microbes that secretes, and then how they works on different cell types within different patients?
Yeah, so how do you start to penetrate the microbial world and its products, its goods and services into a better understanding of cell biology? A kind of cynical way of answering that question is in a few years' time, that'll be up to all the people that are in the room that are a lot younger than me to work that out and how to do it. Okay? But what we're trying to do at the moment in a rudimentary way is, you've seen us try and use in vitro mammalian cell cultures and look at epithelial biology outputs in relation to challenges with different microbes, both thought to be beneficial as well as detrimental. Okay. We've progressed to using organoids rather than a singular cell-based approach to try and achieve that. We've been doing it with organoids derived from duodenal tissue, for instance, and we can show that exposure of the microbiota to different types of food additives can alter the way that community engages and interacts with outputs from those spheroids as a result. Okay. So I think the other technology advance that's taken place here is in recent times that we're currently not using but is highly valuable potentially are these so-called organs on a chip associated with microfluidics. So you could envisage how you might be able to use secretomes from individual microbes here or from a consortia, and with that microfluidic purpose and an organ on a chip sort of model, and there are some for colonic tissue as well as for duodenal tissue, start to monitor how that cellular response is influenced by exposure to these different products. So I think, we've moved. I guess where I stutter a little bit is because I think of what is being referred to these days as multiomics, okay? And I think of the omics of that part of multi, okay? The big difference now is the technology and the instrumentation and the scale that it could drive, okay? But these approaches to look at small molecule analysis and use LCMS or GCMS to try and identify bioactives and through small molecule analysis. I was doing that when I was a PhD student. So in some respects, I was doing metabolomics research back then, but within the framework of a very narrow scale and scope of being able to do it, whereas now it's changed dramatically. And that's the exciting thing for early and mid-career scientists, is having that holistic information to work with.
Yeah, so sometimes we get to ask about a customer come to us and let us which one is better, whether 16S, amplicon, or metagenomics will suit their research objective. Do you see there's a huge difference as improvement in terms of taxonomy and the functional characterization between using the metagenomics, like shotgun metagenomic sequencing, compared with just the amplicon sequencing?
Yeah. Short answer is I would use a shotgun metagenomic approach for just about any sample that I want to really characterise. But really, at the moment still, even though people are trying really hard and making some progress to subtract away what might be considered host-derived genomic information, it's still cumbersome. And so if money is no object, ultra-deep sequencing on samples where there's lots of host DNA as well as microbial DNA, you can get away with it. But 90% of the labs don't have that funding level, and the other 90% don't have any funding at all. So, so taking that approach is hard. But if you have if your sample has high microbial biomass with limited contamination, shotgun sequencing as deep as you can afford is the most viable way to go forward. Okay, we still use 16S sequencing, but where it's being used for profiling is because when it's a little bit cheaper per sample, and you want to almost like screen the population. Okay, tools to infer function from community profiles is starting to, have been around for a while, but they're getting better all the time. The other thing that's happening is that when I first started in this space, we used to make clone libraries of the ribosomal RNA gene, or the gene encoding 16S ribosomal RNA, and we'd make clones of the full length of that gene, okay? And if we could afford it, we'd sequence about 100 of them. And then we'd say we've got a very good assessment of the microbial diversity, okay? Where the sequencing techniques were was through short read technology. It really just scaled up the number of samples and the amount of data you could produce, and it was fantastic. But the challenge with short read sequencing technology is that the confidence you can have in the speciation is good, but it's not as good as it can be using some of the longer read technologies that are available now, and also shotgun metagenomic sequencing, where you don't have to infer anything. Okay. The last point is PCR is biased. Okay. Whereas presumably with some of the library construction that takes place for shotgun sequencing and the way the libraries are constructed, it's non-biased. So you won't have that influence of your PCR primers or your conditions on the sorts of data you make. So I go with metagenomic sequencing for as many of my samples as I could, but our go-round is, rather than doing 16S profiling, is use our skills and knowledge about how to culture microorganisms from tissue, enhance the size of that microbial population, okay, and then go to Novogene and ask for 10 gigabase pairs worth of sequence for the mucosa-associated microbiota.
I think that is a great last question. Please join your hands and thank Mark for joining for the beautiful talk.
So before I want to start, I want to acknowledge that this perioperative trial that I'm going to talk about today wouldn't have been really possible without a weird leadership structure in our lab that involves Jim Whittle, who is a neuro-oncologist, and Sarah Best, who is a cancer biologist, as well as really the leadership of Professor Kate Drummond, who is the head of neurosurgery at the RMH. So as was introduced, we work on brain cancers, and particularly gliomas is our interest in the lab. And recently we've had really an interest in those gliomas that have an IDH mutation, because there has been a new therapy that has entered the clinic recently against this particular IDH mutation. However, what we do know is that most people that do get this therapy actually will ultimately progress. And why that is actually very poorly understood. So the perioperative trial that we were able to conduct here is going to really allowed us to actually identify some of the resistance mechanisms that come into play when we inhibit the IDH mutation. But first, let me tell you about gliomas. So gliomas are really characterized by this diffuse infiltration into the normal brain. And this is a property that makes them really impossible to cure. So the normal treatments that people go through are normally a surgical intervention, and then some treatment strategies, and we'll talk about the IDH mutation inhibitor. But all gliomas ultimately recur. And this is really best illustrated with this case example of a 37-year-old whose cancer was removed only in 2019 to be back then just less than a year later, and then again slowly really grow back. So patient survival for glioma is abysmal poor, with an average 5-year survival rate less than 20%, and that's a lot poorer than what we typically see for cancers in Australia. So as I've kind of hinted, and adult gliomas are classically divided into 2 subgroups, and these are based on the presence or absence of this IDH mutation. In IDH wild-type cells, so this is your classic glioblastoma, isocitrate is catalyzed into alpha-ketoglutarate. However, the presence of mutations in IDH1 or 2 really lead to the production of 2-HG, so 2-hydroxyglutarate. And 2-HG has pleiotropic effects within the cell and the microenvironment that drive tumorigenesis, and that includes oxidative damage, dedifferentiation, and then tumor growth. So like for all brain cancers, the tumors that grow as a result of these mutations consist really of multiple cell states mimicking those found in the normal brain. So as it stands, there are 5 main malignant cell states that we can see here. So a MES-like state, an NPC-like state, an AC-like state, an OPC-like state, and a progenitor population, and that acts as a cycling population that can transform into any of these other glioma cell states. So I just want you to keep that in mind, because we're going to be talking a lot about these cell states today. And notably, what we see is that most gliomas show really a composition that includes nearly all of these cell states. This is actually glioblastoma, and you can see how very diverse the tumor microenvironment is. Overall, patients with heterogeneous tumors really have poor survival outcomes. And as a field of research, some key questions remain unanswered. And those are, how does tumor heterogeneity impact treatment response? And how does this landscape evolve in response to treatment and then lead to resistance ultimately? So now let me tell you about a new treatment that is available for glioma patients with an IDH1 mutation, and that's this mutant IDH inhibitor developed to target this key oncogenic driver. And in the Indigo study, this is a landmark clinical trial that was published in 2023, they really showed that giving patients this inhibitor really led to a significantly improved progression-free survival and delayed time to next intervention. And this is great news for patients, but virtually all patients ultimately actually relapse on this inhibitor. So it gives them more time, but it's not the cure that we had hoped it would be. And this suggests that the tumor cell heterogeneity really leads to adaptation to this mutant IDH inhibitor. But we really don't know how this actually happens and what we would do in order to stop it from happening. And we believe that really only perioperative trials, and this is where we get tumor tissue before and then crucially after a defined window of treatment with this IDH inhibitor can really directly shed light on the treatment mechanism. And such trials would really allow us to observe in real time and in the same patient how tumor cellular landscapes respond to therapy. And this is an approach that is now really gaining traction, led really from Australia, with multiple international groups looking to embrace this framework. But we were really the first ones to do this in Australia, because we had generous funding from several philanthropic, government, and industry partners to really pioneer this approach in brain cancer. And let me just say again, this is very difficult in brain cancer because people do not want to undergo 2 very invasive surgeries typically. But we were lucky to find 10 patients that enrolled in this first clinical trial. And they underwent a biopsy first to confirm their diagnosis of an IDH-mutated glioma, and then they had 28 days of treatment with an IDH inhibitor called safusidenib. And then after this round of treatment, then patients underwent a gross maximal tumor resection. And the collected matched tumor samples were then used in a range of downstream translational analyses. And these ranged from really straightforward ones to much more complex ones involving single-cell transcriptomics of the matched tumor material. And this was where we particularly utilized Novogene and really kept cost low by using them. So the first thing that we did in this clinical trial was to actually confirm that safusidenib indeed reached the tumor and had on-target effects. So we detected safusidenib in therapeutic concentrations in the plasma as well as in the resected tumor tissue, but not in the CSF, and found that these concentrations that we saw in the tumor tissue were directly correlated to the ones we saw in the plasma. So I told you about 2HG, which is here being targeted, and we found that we saw indeed this reduction in 2HG that we were meant to see after we were treating the patients, and this was in the range of 88% across all our patients. In collaboration with Bio21, actually here, we also did spatial metabolomics on a couple of patients and really saw that there was indeed like uniform reduction across the tumor tissue after giving patients safusidenib. So in addition to HG, we also confirmed expected secondary effects of mutant IDH inhibition, such as altered abundance of other metabolites in the Krebs cycle. You would also imagine that there would be effects on the methylation because 2HG reduction is linked to global methylation levels, but we could not see that here. And we couldn't see that globally or locally. So that meant we went to histone modifications. And while these were not significantly altered, we did see trends, in particular in H3K9 and H3K27 monomethylations, where we saw a reduction after treatment. But interestingly, these came with a concatenant increase in acetylation, suggesting that there was global transcription that was altered post-treatment. So to look into the transcription in more detail, our really talented PhD student, Montana, then looked at the single nuclear RNA-seq data from 9 patients which matched pre- and post-treatment samples. So you'll have noticed that we dropped 1 patient, and that was because we had unfortunately sampled normal tissue rather than tumor tissue. And comparing these matched tumor cells, Montana conducted a gene set enrichment analysis and found, contrary to reports that had previously shown an AC-like increase post-treatment, that we actually found an OPC-like and an oligodendrocyte and OPC gene set enrichment. And this was in expectation that we saw these differentiated cells, and these were when we split the tumor cells up into their further compartments. So when we split the tumor cells up into their further compartment, we really saw that we saw this increase in differentiated programs as expected in the AC-like compartment as well as in the OPC-like compartment. But something different was happening to our progenitor cells, which really interested us. So looking at these cycling progenitors, we found that the mutant IDH inhibitor treatment in this population really strongly upregulated this OPC-like set, which was driving the signature you had seen on the previous slide. And not only was this population significantly it was also the only population which had significantly differentially regulated genes with bumper and MERV, also hinting again to this oligodendrocytic function that we were seeing increased. And this really sparked our interest in this population in particular. And so we applied CytoTrace2 to our data, where we were able to demonstrate that this population had a greater differentiation potential post-treatment, and that was different to, for example, the AC-like population. And then next, we asked whether this greater differentiation potential could also be translated to a greater heterogeneity that we were seeing with respect to these other cell states that this particular cell cycling progenitor can take on next. So for this, we projected the progenitor cells into this two-dimensional butterfly plot that is very commonly used in glioma research, and looked and so where every corner represents really one of these cell states. And this really showed us that there was greater heterogeneity post-treatment. So together, what we believe here is that this really hints at that progenitor cells are the reservoir for cellular adaptivity in response to mutant IDH treatment. So the shift in progenitor cells towards this OPC-like state suggested to us that cell states might be involved in the response to mutant IDH inhibition. And given that the IDH enzyme facilitates alpha-ketoglutarate out of the Krebs cycle, and mutant IDH inhibition increases flux through the Krebs cycle, we hypothesized that differential energy consumption through the central carbon cycle may be dependent on certain tumor cell states. And to investigate this hypothesis, we turned to an independent untreated cohort, also generated with single nuclear RNA-seq data and also sequenced at Novogene. And deconvolution of the cell states in this dataset identified really a clear enrichment of a glycolysis signature in normal astrocytes and AC-like tumor cells, and that was vastly distinct from what we were seeing in the OPC-like tumor cells. And this can be a little bit better appreciated, this shift between sort of lactate, glycolysis, and Krebs can be a little bit better appreciated on a ternary plot where you can really see that shift between those tumor populations. However, these observations were merely based on transcriptomics. So we wanted to go a step further and really confirm that this is true by directly measuring metabolic activity. So for this, we turned to a workflow developed by one of our postdocs in the lab, an imaging specialist, Jürgen. And what he had done was collect sequential slides from IDH glioma tumors. And these sequential slides were either going to H&E or spatial transcriptomics with immunofluorescence afterwards, or spatial metabolomics. And for this, he had to achieve multiple techniques to allow us to do this, because we wanted to do this on frozen tumor tissue. So he had this clever idea of first generating a micro CT scan, so that would allow us to really optimize the cutting window for these slides. So they would contain the fewest tears and would allow us to align these slides later on. So spatial transcriptomics here was done using the 10x Xenium over at the WEHI with 330 custom gene panel. And so after the acquisition of the spatial transcriptomics, we then, as I said, did some post-staining with about up to 8 antibodies, which really allows us to visualize exactly where each cell is and allows us to do cell segmentations. However, this is done on a different microscope to the one that the native Xenium instrument comes with. So what we had to also develop was an alignment strategy that would allow us to put these different put the spatial transcriptomics on top of the spatial metabolomics, to put on top of what we were seeing with the microscope from the immunofluorescence. And to this end, Joel and Jürgen really developed a new pipeline called SMINT that has all of these steps really in one neat package. Analyzing spatial metabolomics and spatial transcriptomics together is actually quite challenging. So not only did we develop a tool for allowing us to align and do cell segmentation, but we also built yet another tool in collaboration with Quan over in Queensland to actually do the downstream analysis of these modalities at the same time. And for this, we built SPA-MTP, which allows processing of the spatial metabolomics, including annotation of metabolites in spatial metabolomics generated from TOF instruments, and then do the combined analysis of the spatial transcriptomics and metabolomics. So it provides options like multimodal clustering, pathway analysis, and has really got some beautiful visualizations that you can do with it. All right, so with all these tools introduced, let me remind you again of the hypothesis we were actually trying to investigate here using these sequential slides. And that was that AC-like tumor cells preferentially use glycolysis, while these OPC-like tumor cells really were favoring the Krebs cycle and lactate. So using the generated and aligned data, what we then did, because unfortunately spatial metabolomics isn't quite single cell resolution, unlike our Xenium, is actually use bins that we were going to shift across the tissue and just count the number of AC-like and OPC-like cells in these bins, as well as measure what the abundance of any given metabolite was. And we can then correlate these two things. So, and then this really confirmed to us that there was an increase in abundance of alpha-ketoglutarate in regions that were really rich in OPC-like cells, whereas in these AC-like rich regions, we really saw glucose as abundance. So next, we hypothesized that the difference in metabolic capacity might be involved in the efficacy of the mutant IDH inhibition. So given that the clinical data from our trial is really yet to mature, because patients were only treated in 2022, we turned to published literature, which so far really only contains a single responder and a single non-responder profile pre and post mutant IDH inhibitor training in using the single cell transcriptomics like we had done. And using this cohort, we found that enrichment of OPC-like signatures in the progenitor tumor compartment was really associated with non-response to mutant IDH, while the upregulation of the AC-like program in this compartment was seen in the responder sample. And so these findings really enabled us to postulate which patients in our cohort are more or less likely to be responders. So hence we kind of have a foundation of a possible biomarker for mutant IDH inhibitor response here. This is cool, but we totally agree that this needs confirmation. Obviously, our clinical data will either confirm or not confirm this. So we'll update you in the next in the coming years. But next, I wanted to really turn to one of the other very striking results that we had from our single nuclei analysis. And this was the downregulation of neural synapses-related gene sets, and that was exclusively seen in the neuron population post-mutant IDH inhibition. In fact, all the other populations showed an increase. So to make really sense of this result, let me introduce you to cancer neuroscience, which I think is a really super exciting field. In the past 5 years, people that were looking at this have really shown that glioma tumor cells form synaptic connections with neurons in the immediate environment. And they exploit these connections to use neuronal activity to promote their own growth and invasion capacity. So that's quite sneaky and obviously also really hard to treat. At the same time, what we also know is that 2-HG is known to enhance neural excitability in IDH mutant glioma models. And this excitability can be reduced upon mutant IDH inhibition and associated 2-HG depletion, but that has only ever been shown in a mouse model. It may not be surprising with all of that said that the latest updates from the Indigo study really indicated that there was a significant reduction in seizures reported in patients taking the mutant IDH inhibitor versus those that were on placebo. So it made sense to us that we were seeing these results given all of that and that they could be beneficial to our patients. But we wanted to see whether we could see additional data that really made sense of sort of the mechanism behind it and what was really happening here. So we turned to some of the other data that we had collected, in particular the spatial metabolomics and the metabolomics itself, where we really identified a reduction in neuromodulator adenosine as well as pathways related to synaptic signaling post-treatment. However, none of these analyses could really show us whether we were seeing this reduction due to a systematic difference in composition between the tumor that we sampled at biopsy versus the one that we sampled at resection. To overcome this possible sort of bias in the microenvironment, we actually looked at our spatial transcriptomics data, which we had collected from 3 patients. And what we did here first was identify microenvironments that are similar between the samples. And we did this using GraphSage. And what you can see here is that we have one sample, 001, that had the same dark green microenvironment in both the biopsy as well as the resection. And this dark green microenvironment is interesting because this is the microenvironment that contains neurons. Around 25% of its composition is neurons. We further confirmed that indeed this is actually a leading edge where the tumor moves into the normal using markers of the normal brain layers here. And then we use differential expression analysis of synaptic signaling resolved by different neuron types, including those in layer 2, 3, 4, and 5, 6, as well as inhibitory neurons to clearly demonstrate that synaptic signaling is really reduced post-safusidenib or post-treatment in all these neuron populations. We also could look at some specific genes and really show that decrease for this, admittedly just one patient. However, what we also we were fortunate enough to do was actually collaborate with Lucy Palmer over at the Florey, who had fortunately received live, very fresh tissue from those patients and had captured via cell patch clamping electrophysiological recordings of the neurons. So she'd received ex vivo slices from 7 matched patients and was looking at layer 2, 3 pyramidal neurons. And she could show that there was a dampening of the overall excitability in response to step injections, as well as reduced firing rates. And this really confirmed that the electrical environment is altered following mutant IDH inhibition. So with this, I just want to close and say that perioperative approaches are really safe, feasible, and acceptable for patients with brain cancer. That this platform that we have here in Victoria and is the first in the world really represents an opportunity for drug development. And as a result of this clinical trial, we have seen a lot of interest from pharma and are doing about 4 more of these types of trials. And single cell analysis was able to demonstrate in this particular case that transcriptional changes characteristic of differentiation as expected were happening, but that one tumor cell state, the progenitor cell state, was quite different, displaying this increased cellular adaptability that might be involved in the resistance to the mutant IDH inhibitor. And that this is linked really to the metabolic remodeling that is happening post-mutant IDH inhibition. And finally, possibly most excitingly, we really showed that safusidenib had these off-target effects that really reduce neuronal excitability that could be quite helpful for the patients. And with this, I'm going to finish and say for something like this to work, it really needs a very large team. So there are lots of people to thank, but I just want to particularly shout out the people in this building here that made the spatial metabolomics possible for us to do. So thank you so much for your attention.
I think I'll kick off the first question. I'm just wondering, because as a scientist, you develop so many new technologies and different platforms, and I'm so happy to see you got your paper accepted in Nature Medicine. But have you had any challenges into like convincing other fellow scientists that this is the right workflow? And have you had any challenges into publishing to these journals?
So first up, we were really fortunate to have a great clinical team that was really understanding the value of translational work, and we're happy to throw everything at this study. This being the first of its kind, we kind of wanted to try everything. In follow-ups, the next couple of studies, we're not going to do this very vast set of different technologies, but probably pick and choose our technologies a little bit more, given sort of what makes sense with the hypothesis that we're going for. Mutant IDH inhibition is obviously really interesting because there's a lot of different things involved, from metabolomics to epigenetics, really sort of everything. With the journals, it was quite interesting with Nature Medicine. I guess the process was very much the science first and technology later. So the first couple of reviews we had, the technologies weren't really mentioned, and then we had a technological review right at the end. And well, I was lucky to say that it just went flying through. So very happy with that.
Well, I'm just curious about the first part of the story, bit confused. So whether the treatment has been given post-surgery or pre-surgery?
So they have two surgeries. The first surgery, and both of them are open craniotomies, which makes this kind of confusing. In the first open craniotomy, the surgeons only take a small sample, the biopsy sample, and then right after that, the patients start safusidenib, and they do that for 28 days. After 28 days, they actually take their final dose on the day of the last surgery. And then they have a normal gross total resection, again with an open craniotomy. And at that point, the surgeons, like normal, try to remove all of the cancer. And that gives you the ability to look between the biopsy and the resection of whether there is a difference.
Follow-up question on that. Whether the drug has been injected in CNS or in the IV?
This is an oral medication. Yeah.
Because you're mentioning about it is a kind of inhibit the Krebs cycle once. So it could be a possible lot of off-target effects because Krebs cycle is very important. Did you see any serious complications in the patients?
So we did report on the complications as well. There weren't any severe complications in this one. I think after the study, after this first part of the study completed, I think one patient has come off due to adverse events. Well, not they didn't want to continue, basically. Yeah.
Yes. Any more questions? Thanks, Saskia, for the useful talk and this. It's also exciting to see how the data we from Novogene has been translated to some exciting findings. So you mentioned a bit about the challenging of really integrate and normalize data from different omics. Do you have any suggestions for researchers who want to combine multi-omic research, starting from sample collection to project design to get this process easier?
Yeah, I think what is really important is thinking about the different requirements for your multi-omics assays right from the start. because if you're collecting, if you put everything into FFPE, then you're not going to be doing metabolomics. So and for us that was one of those things where we had to, you probably have to be quite flexible in a study like in a clinical trial like that with what you can do and what you can't do, and then optimize for what you're trying to achieve. And I guess it's much easier to do this in a non-clinical trial setting, as the one with the frozen tissue where we did spatial metabolomics and spatial transcriptomics from sequential sections. We wouldn't have been able to do that in a setting like a clinical trial, just because logistically that is so challenging to do, and then putting that on top of the logistical challenges of a clinical trial, that's just not really feasible. So you kind of have to think around those ends a little bit. And then I guess the other thing is all of these different omics technologies take a lot of tissue, right? And that's really a limitation when we're talking about things like a biopsy sample that is really small. So we had to we were fortunate enough that we got enough tissue here for everything, but that's not always going to be the case down the line. And certainly we don't have much tissue left now for further investigations we would like to do. So thinking about what you really want to do up front is very important.
Just a question about brain. So, like in immune studies, the drug, the cells die and go away, and you have brand new cells. Are these cells in that area just always there? They survive and they live there?
You mean immune cells?
No. In your cross sections, so the pre and post sample.
Yeah.
Some cells die and get replenished. And they wouldn't get.
So in terms of proportion, yeah, we didn't. Proportion is really hard to assess because of the systematic sort of bias that you have. Looking, I unfortunately don't have it in here, but basically looking at sort of the pre versus post proportions, we had the feeling that we were getting quite different things, and that was a systematic bias rather than something that that was due to the treatment. It was probably because the surgeons took out a piece that was probably closer to the edge, and then post it was actually closer to the middle of the tumor. And that's obviously systematic bias. And as I've said, we try to overcome this a little bit with spatial transcriptomics in particular, because it's like so hard to control for. And I wish, what I really wish is that our surgeons would be able to give us location, which I think in future trials, there have been plans in future trials to actually do that a bit better.
All right. Well, thanks everyone for coming. Thank you, Novogene, for the opportunity to present our work. So I was going to give like two short summaries from two different sort of projects, but just to highlight the different techniques we use since we're going for a bit of this multi-omic angle. So every cell in our body has the same DNA sequence, right? So that's the basis of that every cell is the same. And we have about 2 meters of DNA that has to fit into a tiny little cell nucleus, right? So it really needs to be compacted really tightly to achieve that. And each level of this compaction, as you go from a very compact chromosome down to DNA, is you can have different marks added to the DNA. So these are called epigenetic marks, and you can either add them to the histone tails, or you can covalently link them to the actual DNA itself. And then from this one sort of DNA sequence, you can get lots of different cell types, right? They all have the same DNA, same genome, but a different epigenome. And the efforts to understand the sort of genome, well, more or less concluded in 2001 in the sense that we sequenced and had a sequence of the genome. But only recently did we start to sequence sort of more diverse regions of the world. So that's still ongoing. But as early as 2010, we knew that this wasn't everything, right? That there was a layer above the DNA that sort of complicates things, that makes things more interesting. And sort of the effort to understand the epigenome is sort of loosely under this umbrella of the International Human Epigenome Consortium. And over the years, there have been many different studies all across the world looking at analyzing the epigenome and characterizing it. So my sort of interest in the sort of epigenomics and multiomics came from doing a postdoc as part of this Blueprint project in the Netherlands. And what we were trying to do is make reference epigenomes of every cell type in the blood. So what that means is sort of applying different techniques to understand different aspects of the epigenome. So including looking at gene expression, looking at where the open chromatin was and the heterochromatin was, looking at different histone marks, and looking at DNA methylation. And in particular, what I'd like to talk about today is histone marks. So what we call the reference epigenome was like a comprehensive map of all epigenetics in the cell. That sort of tells you something about the cell, right? Whether it's something about its past, like exposures, or its present state, or what it can do, like how it can respond to stimuli, for example. And while the histone code has over 60 different histone modifications, by generating about 5 of these using ChIP-seq or different techniques, you can sort of use them as markers for the rest, and you can get an idea of what's happening in the cell. So I'll give you an example of how we use this in the context of trained immunity. So this is funded by various funders, and I've been working in this field for about the last 10 years. So what is trained immunity? It's a innate immune memory. And classically, when we think of the immune system, we have the innate arm, we have the adaptive arm, right? And the main difference sort of that you get taught is that the innate arm has no memory, and the adaptive has memory, which is genetic, specific, and can last for years. And when we sort of think what happens is the innate immune cell comes along, it finds a microbe, it eats it, and then it shows the antigens to the adaptive immune system, which then generates a memory. And then this cell, we thought in the past would just die, or new cells come around and they do the exact same thing again. But what trained immunity tells us is that these cells, the innate immune cells, like monocytes, for example, internalize these inflammatory signals and then show stronger responses to unrelated pathogens. And this is all wired epigenetically. So basically, there is a memory, it's epigenetic, it's not specific, and it can last for weeks to months. And we see it in some sort of off-target effects of vaccines. So over the years, we've shown that this can be sort of induced by all sorts of different stimuli, like microbes, danger signals, vaccines, and metabolites. And you get this flavor of different macrophages. Some are less responsive, some are more responsive, all due to the fact that they were being epigenetically and metabolically reprogrammed. So you can do this in vitro. So if you like this method, because it's sort of simple to do, you can do it on primary cells. You get monocytes out, you stimulate them, you let them rest, and they differentiate into macrophages, and then you re-stimulate them with something unrelated to the first stimulus. And we sort of characterize it by how much cytokines they can produce. So if you stimulate a cell with a trained immunity stimulus, you'll get stronger responses when you expose it to something else. And for example, if you tolerize it, like you see in sepsis patients, they're unresponsive. And all of this is epigenetically mediated. So one thing to think about if you want to do epigenomics, you're thinking like what you want to apply and when, is that epigenetic marks have different half-lives. So some things happen really fast and then they go away, and some things take longer to be established. So if you think about time on the x-axis, and this is the signal in the cell, in response to any stimulation like a vaccine, you'll get gene expression going up, an active histone mark, for example, H3K27 acetylation. And it's only after these marks have been established and are leaving that you start getting the priming histone marks and finally DNA methylation, which can last for many years in humans. And all of this reprogramming changes how you respond to the second exposure, right? So you have this trained response because of all these events that happened prior to it. You're basically shooting this cell off a different trajectory and it's responding differently. So here's an example from a study that we did a while ago. What we did is we took a monocyte and we gave it a lot of LPS, a lipopolysaccharide. Basically, we tolerized it. We gave it so much that the cell responded really, really strongly, and it looks like a sepsis-like tolerant cell. And then we generated 5 different histone marks, so 3 active marks and 2 inactive marks. And basically what I'm going to show you is that by using these different marks, you see that all of them are changing at the same time together, right? So here's an example of a gene. This is IDO1. It's important in tolerance. And about 10 kb down upstream, there's a enhancer. And you can see at baseline, we're looking at just K27 acetylation, K for mono, there's no signal. And if you stimulate these cells with LPS, you'll see that you're now activating this enhancer region. You're basically opening up the chromatin. But if you look at day 6, after 5 days of rest, and you were to look at acetylation, you will see nothing there, right? So it did its job and now it's gone. So if you were to look at acetylation at that point, you would not see it. You would not see any sort of difference between a tolerized and a normal macrophage. But you do see the K4 monomethylation as a memory. So what we can do is we can sort of separate the genome into like different states, depending on these 5 different histomarks. And some of these states, we don't know what they are. They just got everything. But basically what we can see is that this region was actually repressed by K27 trimethylation and had a little poise enhancer. And when you stimulate with LPS, you're not only activating this region, you're also removing the inactive marks, right? So it's all happening at the same time. And another thing that's sort of interesting is that we have these 2 sort of things happening in training immunity. We have the epigenomic remodeling, we have the metabolic reprogramming, but they're actually quite intimately linked. So in 2019, there was a new publication about a new histomark called histone lactylation. And it's added right here on histone 3, lysine 18, right? So it's a new mark. So it's an evolving field. Basically, we can find that most things can be attached somehow post-translationally to histones. And what we did is we looked at this histomark in the context of trained immunity. So I'll just quickly go through these results, just to give you an example of what that looks like. So what we're showing is the metabolic reprogramming is directly incorporated into the epigenome. So what happens in these cells, so similar to what Saskia was talking about in cancer, is that they show the Warburg effects, right? So they go from oxidative phosphorylation to glycolysis. They stop using oxygen, they start using anaerobic respiration. And what we think is happening is they're basically just upregulating glycolysis to such a high extent that they sort of look like they're very strong and primed for responses. So if you take an in vitro model and you stimulate cells with this BCG vaccine, you have more lactate production at day 6, right? So you've induced this trained immunity phenotype. And what happens as well is that this lactate gets incorporated into the chromatin, and you can see that globally there's more lactylation in these cells, right? Not just at a specific site. So what we did is we did ChIP-seq for HVK18 lactylation and RNA-seq, and just to basically show that this mark more or less acts like an active histomark. It's very similar to acetylation in terms of where it's deposited and how it correlates with gene expression. And what then you can do is you can block the production of, you can actually block glycolysis by using these inhibitors. You have less lactate production in these cells. And you also see here, we're looking at histone marks at a promoter region. You can see these 2 peaks, that's where the 2 histones are sitting. And this is right where the gene transcription is starting. And you can see if you block LDH, you start to get slightly less lactylation in these BCG cells. And because lactylation is added by the same histone acetyltransferase, P300, if you block that, you also completely lose the lactylation. And then what happens, because we know that the genome is in a 3D conformation, right? So promoters can touch each other, enhancers can touch promoters, right? What we can then show is that at this R1 beta locus, which is very, very well characterized in trained immunity, here is the gene, and all of these enhancers here are touching that promoter, right? So they're in a 3D conformation with that. And if you stimulate cells with BCG, you get more lactylation. And if you block the production of lactate, you lower these connections, right? So it kind of just shows you that affecting one thing, part of a cell, has all these downstream effects, right? And they're all sort of linked, and they all sort of control how genes are expressed and how cytokines are produced in these cells. So I'll just like to summarize that, even when we talk about a single sort of omic, because people always come up to me generally and they'll say, I want to do some epigenetics, what should I do? You have to sort of think there's many different components, right? So every field is complicated if you're into it and you do a lot of work in there. And then profiling these marks in combination, if you have sort of a nice functional outcome, or sort of like you say, like a nice clinical trial where you know that there's a well-defined reason for doing something, applying these techniques can actually add a lot of interesting data to it. So now I'd like to sort of shift and talk about what we're doing at the moment in human cohorts. So at the MCRI, we have access to a lot of childhood cohorts, birth cohorts. The data I'm going to present today is from an adult cohort. And what we're slowly, and I guess most research institutes and most teams are going into this direction, is that you're trying to move from your sort of area of expertise or sort of specific area and try to do many different things with collaborators and clinicians. So this work is funded by the Paul G. Allen Frontiers Group, and we have another 3 years left of this grant. So what we're trying to do is we're trying to understand how sex hormones affect the epigenetics and immune function in humans. And we're using gender-affirming hormone therapy clinical trials as our tool. And there's a lot of people involved in this research, both endocrinologists and gender-affirming hormone therapy sort of specialists and molecular biologists. So why are we studying it? So the sort of primary question when we started this work was that there are sex differences in immunity, right? Well, across many human traits. So bone density, muscle mass, different disease risks. And this is also true for immunity. So I just like to point out, for example, when we look at autoimmune disease, some of these, like SLE, like 90% of the incidences are in females, right? So there's a really big skew towards female in autoimmune disease. But if you look at outcomes sort of of infection, males seem to respond worse, right? So we have this imbalance. These females that respond better to infection, but they're also much more likely to develop autoimmune disease. And the question is, can we differentiate between the influence of genetics and sex hormones on these disease risks, right? And in general, the answer is no, because they coexist, right? Early on. Of course, there are methods you could do it. So gender-affirming hormone therapy, about 0.6% of the population. If you look in Australia, the latest from the Bureau of Statistics is about 1% of people 16 and over identify as transgender. So now that's quite a big chunk of the population, and 90% of them are probably going to start gender-affirming hormone therapy. So in the US alone, that's about 1.5 million individuals, about 200,000 in Australia. And the primary aim is to alleviate dysphoria around physiological changes. So the primary outcome is always to see a change towards the opposite sex. But the long-term consequences are unknown. So no one's really been studying in this field. It's been a bit taboo. It's not really, you can't study in certain countries. You can't do it at all. But basically what it looks like is if you are a participant in a feminizing gender-affirming hormone therapy, you will receive estrogen and anti-androgens to lower your testosterone. And if you're receiving masculinizing gender-affirming hormone therapy, in transgender men, for example, you're receiving testosterone. And we know from studies in Amsterdam over the last 5 decades is that transgender individuals live shorter lives, in particular transgender women. And this is sort of showing overall survival over 3 decades. So the mortality rates are much higher due to HIV, which is 15 times higher, suicide 3 times. So these are unrelated to the actual hormone therapy. But what's interesting is that cardiovascular disease and cancer rates are much higher. So those sort of things could potentially be a result of receiving treatment. We also know there are higher rates of smoking, drug use, and there's also hesitancy to see healthcare professionals, right? So just from that perspective, it's important to include them in research just to get them into the medical system and get them sort of comfortable that people are here interested in what happens to their bodies. So we did a study, and this also, I guess, is a good example of what Saskia was talking if you're planning a research study. So we had clinicians, and they said, we started a new cohort, but before that cohort, sort of got up and running, they said, we already collected some samples in the past from a hormone trial, but it was basically just blood and it was just frozen down. So all you could really do is methylation. There was nothing you could really profile in those cells. So we thought, that's a nice way to start. So we had samples from a feminizing trial and a masculinizing trial at 0, 6, and 12 months. So these are longitudinal samples. And we had whole blood frozen. So what we did is we did an EWAS using an EPIC array. So it's a methylation array that doesn't look at the whole global methylation, but looks at specific sites. And what's kind of interesting that we found was first that feminizing and masculinizing gender-affirming hormone therapy induce their own DNA methylation profile. So they're not like opposites. It's not like one goes this way and then the other one goes the exact opposite way. So if you look at these regions, for example, and here I'm showing DNA methylation, here I'm showing transgender men receiving testosterone over 12 months, and here are transgender women. So for example, you can see these regions are gaining methylation, and these regions are losing methylation if you're receiving estrogen. But they're basically more or less static in the transgender men. And vice versa, if you find regions that are changing in response to testosterone, they're not necessarily changing in the estrogen. And this is an example of one locus. So here, these individuals are receiving testosterone, and here is a gene. So this is the promoter. It's got this ENCODE DNA peak, so we're sort of looking at where the open chromatin would be. And we have a differentially methylated region right here. So what I'm plotting here in black is an assigned male as a control. These are the transgender men baselines in the light blue. And you can see that around this gene, basically, there are no differences between sexes until we get to this differentially methylated region. And this is where it's sex-specific. And if you receive testosterone for 6 or 12 months, you see that this is exactly where you're losing your methylation, right? So it's responding at that locus. Now, this locus is actually a sex and age-specific locus. And what that means, and it's another cohort of about 200 people, we looked at matched Guthrie cards and then adults. They're here about 30 years old. And you can see that if you look at this particular CPG site right here, and you look at Guthrie cards, it's the same between males and females. There's no difference. But if you look at adults, the males have lower methylation, right? So this is something that's most likely occurring at puberty, right? It's being established over time in response to hormones. And that's exactly right. There was a study that showed that this particular site starts to be hypomethylated in males around 16 years of age, right? So what's interesting is that some of our participants are over 65. I think we have 3 65-year-olds and a 72-year-old, and they also see the exact same changes occurring, right? So this is sort of telling us is that humans are pretty plastic, right? You can induce puberty at, in your 60s, at least in these regions. And this is sort of the data from Novogene comes in. What we did since then is we collected some really nice clinical trials. We were able to get some nice funding to do this work. And we just had this paper accepted. Well, we had it accepted, sitting right here in the front row. So what we did is we wanted to look at the plasma proteome in feminizing gender-affirming homotherapy. So we did this in Singapore with Novogene. So why look at the human plasma? It's important because, basically it's something that's easy to get from the blood, and it can be used as a diagnostic or a therapeutic to sort of track therapeutics. And the plasma represents the largest and deepest version of human proteome in any sample because it has both circulating proteins, but it also has tissue proteins. So, right, all tissues secrete some sort of leakage markers that get into the plasma. So by looking at sort of the proteome, we can see what's happening at the physiological level that underlies that. So we use the Explore HT Olink platform to look at 5,000 proteins. And this is a model. So this was a clinical trial done here at the Austin Health. And what's really nice is that because it was a clinical trial, we had 2 very distinct arms. So well, in total we had 50 individuals, but here we ran 20 in each group. So they all received estrogen, but they received 2 different types of anti-androgens, and one of them works much better than the other. So cyproterone acetate or CPA, like sort of lowers the levels of testosterone much more than spironolactone. So this is what happens in their estrogen levels. So for example, this is a baseline and 6 months, and you can see, I think out of the 20, 18 and 17 here reach their target levels of estrogen, but they want to reach in sort of the female levels. But if you look at testosterone, you can see that the CPA lowered, all 20 samples lowered their testosterone to target levels, but in the spironolactone group, only half of them did it, right? So basically what we can now do is we can not only tease apart the genetic effects, but we can also tease apart the effects of estrogen and testosterone. So for example, prolactin is a hormone that's downstream of testosterone. So when testosterone goes down, prolactin goes up. And you can see that this only happens if testosterone has reached its target levels. So using this, in the past, what we did is we showed that we can identify, for example, in this case, metabolites that over time only go down if you're receiving cyproterone acetate, CPA. Whereas in the spironolactone group, even though you're receiving estrogen, it's not going down. So we can then sort of more or less confidently say that testosterone is driving this change, right? Or things that are downstream of testosterone, like other hormones and the loss of muscle and things like that. So we know that sex is a key determinant of the plasma proteome. We know this from UK Biobank studies, but also other findings. And after genetics, basically, so this on the y-axis is x-axis just showing the explained variants in the proteome. These two top bars are genetics, and the third effect is sex, right? So sex is a major determinant of the differences between, or the variation in the proteome, right? So males and females are quite distinct. So we want to answer that question. So we used data from the UK Biobank. They ran 54,000 individuals, and they had a 2,900 Olink platform running these. And they were associated with different biological phenotypes, such as age and sex and BMI. So the question was, could we use our model, our clinical trial, to see how many of these sex-specific proteins are actually influenced by sex hormones, and how many are just unchangeable? So we have 2,600 proteins that are covered on both arrays, both panels. And if you look at the total sort of dynamic proteins, and this is at a p-adjusted value of 0.05, we sort of about 8% are affected by CPA, and about 3.1% of all proteins are affected by spironolactone. So these are the results if you look at the top 100 proteins associated with sex, age, or BMI. So this is our background in our two groups, CPA, spironolactone. And you can see that we're particularly enriched for these sex-specific proteins. So in a way, you can think of this, if you take the top 100 proteins, you say these are different by sex, 35% of them change just in 6 months of receiving estrogen and lowering your testosterone. And if you lower the p-values, and if you sort of just look at the correlation, regardless of a p-value, you'll see that most of them are going in the same direction, even if they're not reaching significance with the numbers that we had. So in essence, within 6 months, you can lower most of the sort of sex-specific proteins just with hormone changes. And for example, here is leptin, which we know is higher in females. It's produced by fat cells, and it's also induced by hormotherapy. So the next question was, what was the broader context, right? Are these changes something that just, is very specific to the transgender population, which is a small population? Or is this something like a fundamental thing that sex hormones do? Can they alter these proteins? So what we did is we used, again, the UK Biobank, and we can link, sort of look at different phenotypes like menopause, menopausal hormotherapy, hysterectomy, oophorectomy, all of which are associated with changes in sex hormones. And this is just showing enrichment. So the gray is a background, and it's just sort of showing in general that all of these different approaches to hormone change are enriched for our gender-affirming hormotherapy proteins. So I'll show you what that means. So what's kind of interesting, if you look at the biobank data, is that you can see if you're plotting estrogen in cis females over time, they're all between 40 to 70 years old, is that if you're not taking menopausal hormone therapy, you have generally high levels of estrogen compared to individuals that take menopausal hormone therapy. But you can see at around 57 years of age, if you are not taking the hormone therapy, your estrogen falls down, whereas this group is low, and I guess that's why they're taking the hormone therapy in the first place, but they maintain that across time. So they're actually higher at later ages. So what we can show here is basically if you're a protein that's being turned off by gender-affirming hormone therapy, these proteins are also more likely to be higher in menopause, right? The opposite effect. But if you're taking menopausal hormone therapy, you're going in the same direction as gender-affirming hormone therapy. So that's in ways telling us that the underlying genetics, there might be some specific differences. For example, NOS3 is only really induced by gender-affirming hormone therapy. It's never positively associated with estrogen in cis females. But it just shows you that regardless of the genetic background on which these hormones are working, they seem to be having the same effects. And sort of that's one idea that we need to disentangle, is like why that might be the case and which tissues or which proteins might be completely refractory to sex hormones. And then another question is, what does this mean for the disease risk? That we can't really, answer this conclusively, but it was nice to sort of just attempt it. So we used data from this paper in the Icelandic cohort, which had both Olink and SomaScan data. And we can look at enrichment of pathways in our gender-affirming hormone therapy. So a lot of these are linked to change in body composition. That makes sense. We have testosterone up there, but we also have things like poultry intake, which at first seemed like odd. It's a bit funny that it's so highly up there, but it potentially could be that there are some things in the chicken and the sort of the way they're treated that might be influencing our estrogen. But it's mixed result. There's really poor papers on that topic. But in general, what we can say is that if you are taking gender-affirming hormone therapy, we're seeing that your protein profile is converging towards that observed in allergic asthma, for example. So potentially you could have high risks of that disease, and it's diverging away from atherosclerosis, which is kind of interesting because it could suggest that, in essence, it might be protective against atherosclerosis, which we actually don't know at the moment. I think there's a lot of literature linking general immunotherapy to heart problems. So in summary, we have several hundred proteins that were altered by feminizing general immunotherapy, involved in all sorts of different things. But what's kind of interesting to me is that, just within 6 months of receiving this homotherapy, we basically changed 8 out of the top 10 sex-specific proteins in the UK Biobank and 35 out of the top 100. So that just sort of shows you that just sex hormone concentrations alone are a pretty big component of what determines your circulating levels, your sort of physiology. So in the broader context, gender hormone homotherapy is pretty similar to the sort of menopausal homotherapy, with some notable exceptions, but the overlap is about 50. So there's lots of proteins that are not overlapping, and it seems that they work the same regardless of the genetics. So what do we want to do? So we're funded for the next couple of years. We got 2 PhD students starting with us. We have teams at the Austin in the Netherlands, at MCRI, just doing different sort of aspects of this. We have a mouse model of gender-affirming homotherapy, which we're not exactly sure what to do with. So that's one thing we have to carefully plan to make sure we make the most of that. What we've generated so far is single-cell RNA sequencing here on campus at MCRI, and that's being analyzed by the bioinformatics team there. We've stimulated these cells with different pathogens, in particular HIV RNA, because of the high rates of HIV. That was one interest of the NIH before the freeze on funding. But luckily, we got the money and we did the experiments before that. And we've also been doing a lot of flow cytometry and trying to see if we can identify if there are differences in cell composition. So I'll end there. I'll just thank all the collaborators. This is funded by not just sort of the NHMRC, but also different sort of philanthropic bodies around the world. And that's sort of what you have to do these days, is try and fund this sort of work. And a lot of collaborators are involved, and everyone's got their own sort of skill set, which, like Saskia said, you can't do this by yourself. And yeah, thank you, Novogene. Thanks for helping, and thanks for having me.
All right. We got a question over there.
Yeah, thank you, Novogene team, to inviting me to give this talk and then share some of my analysis using multi-omics dataset and from the excellent service from their platform. So I'm Claire Sun. I'm the lead bioinformatician here at Hudson Institute of Medical Research in Clayton, Melbourne. We are doing some work to make some breakthroughs in pediatric cancer. So I would like to start by acknowledge the vulnerable people of the Kulin Nation as a traditional owner and custodians of the land on which we meet today, and we pay our respect to their elders past and present. Let me start by sharing some heartbreaking facts about childhood cancer that we need to raise awareness about. Pediatric cancer, although they are relatively rare, is the leading cause of the death by disease in children. And it is estimated by the WHO more than 400,000 children worldwide are diagnosed with cancer every year. And it's definitely underestimated because there are a lot of developed area that we don't have any numbers yet. And we are seeing a continuous increase unless urgent improvements in diagnosis and treatments options are made. And approximately 1,000 of these cases are from Australia and every year, and 3 pass away every week from this terrible disease. So there's no tragedy in life like the death of a child. I think everyone would agree. Every 3 minutes, a child is diagnosed with cancer worldwide. Shockingly, 1 in 5 of these children will not live to celebrate 5 more birthdays, leaving their families devastated. Additionally, even for the survivors, 3 out of the 5 will live in the fear of secondary cancers, infertility, suffer from long-term side effects from toxic chemos, radiotherapy, making their lives a constant struggle. So as researchers, we need to take a closer look into this problem and the alarming stats on childhood cancer. Behind the rising incidence, we see that leukemia is actually the most common form of cancer among children, followed by brain cancer and other forms of solid tumors. In recent years, we have seen a troubling shift in childhood mortality stats. Well, leukemia used to be the most common cause because they are the most common, incident. But because we have made breakthroughs in treating leukemia, now brain cancer has taken over the tragic role, become the leading cause of cancer type deaths in childhood cancer. And it's this one. One promising avenue for improvement lies in the field of precision medicine, as we learned from treating adult cancers, which enables us to pinpoint the genetic changes or mutations that drive tumor growth, and then we can then use targeted therapies to remove or slow down the tumor growth. Current childhood cancer precision medicine program focus on the genomic sequencing technology to very fast and accurately identify the mutations in their genome, trying to predict responses from those targeted therapies that are already established. However, it's unfortunate that children's cancer are known to have a very low mutational burden, as illustrated in the plot here, which compares the number of mutations in children's cancer on the left, the red panel, compared to those in adult cancers on the right. So the low mutational burden means that fewer than 20% of these children harbor actionable mutations that we can have some treatments target. And then among those that actually do, less than half likely respond to those therapies firstly developed for treating adult cancers. So there's a huge gap, and it really underscores the challenges of using DNA mutations alone as a biomarker of response in pediatric oncology, and highlights the need that more research is needed to identify other potential biomarkers that tailored for pediatric cancer to guide the treatment decisions for them. So multi-omics integrative approach has been successfully employed for adult cancers to identify more therapeutic targets. Many of us know, Sanger Institute Cell Line CCLE project, Broad Institute DepMap project, that they easily characterize over thousands of cell line models for different adult cancer malignancies. However, pediatric cancer, especially brain cancer, have been woefully underrepresented in these efforts. The number of disease models to start with is much more smaller, and their characterizations, molecular on different modality, significantly lacks the adult counterparts. So with all this in mind, we started a new program that we call the Childhood Cancer Model Atlas, and we started 2017, 8 years ago at Hudson Institute here in Melbourne, with 3 main goals of program is firstly, take a step back to develop more models, so we can have better genetic diversity to learn to characterize, and then we have a special focus on brain malignancies. And also then to identify new targets, pathways, and biomarkers of response via hypothesis-driven research or more sophisticated multiomics analysis, and then accelerate the implementation of the new therapies, new findings into clinical trials to improve clinical outcomes. So here is basically shows you our multi-omics pipeline, very similar to all other kinds of pipeline work. We not only use established cell line models, we also take tissue from local hospital, from surgery, and then we develop 2D or 3D models in the lab. Then we do whole genome sequencing, RNA sequencing, DNA methylation array. We also now starting profiling 6 or 7 histone markers to get the chromatin states, and also total proteomics, phospho, and maybe some other PTMs adding to this panel. This is the molecular landscape. We're trying to, understand them as comprehensive as possible. And also we do high throughput CRISPR screen and drug screen, which help us to tie the molecular features to the functional outputs. As we enter our 8th year, we have expanded our data set to create a comprehensive resource. We published in 2 years ago in Cancer Center, and then we keep growing now encompassing the world's largest collection, single-site collection of high-risk pediatric solid tumor cell line atlas, which is a true cornerstone of our program. We work day by day to add more models, have more data, and then this portal showing a screenshot here shows you the real life updates of our program, and also you can look into the analytical portal, download all the process data like a pediatric DepMap program here. So to firstly get some idea of the whole data set or some kind of overview, we applied multi-omics factor analysis or MOFA to examine the whole thing in an unsupervised manner. So we don't label any data points. We just feed in the multi-omics data set and then integrate them to see how they cluster. So this basically shows you the analysis. MOFA is really a good tool for this purpose because firstly, unsupervised learning, no per label required. And also it handles missing data because for large scale pipelining work, you just not always be able to fully characterize every single cell line models on every assay. So it's really handy for having the ability to handle the missing data. And also in their dimension reduction analysis, we can also freely annotate the latent factors with known information. So to better understand which latent factors contribute to any clinical features that we know. And then everything will be fed into downstream analysis. As you can see in the final UMAP of clustering analysis, the cell line models now more than 400 now. This only shows 200-ish, I think. All the models after learning DNA, RNA and DNA methylation profiles, they form tight clusters considering all the features according to their cancer types. This really gave us faithful, confidence to know our models are representing the tumor to a very good, faithful level. At the end of the day, they are not real tumor, but we hope they can represent the tumor models to a good extent. So today I'll walk you through maybe 3 of our ongoing projects to show how we use this data set to uncover new therapeutic targets and biomarkers by using multi-omics analysis. So firstly, we use machine learning or AI to integrate multi-omics data to predict gene vulnerabilities we identify through CRISPR and drug response predictions. And then we want to have more ideas for combinatorial treatment. So we also analyze DNA and CRISPR together to find synthetic lethal interactions. And then we extend it to look into more epigenetic therapies. Is there any way we can harness the plasticity of epigenetic landscape there? So firstly, there are many questions that AI nowadays can help us answer in our day-to-day research and clinical work now. I think everyone would agree how lovely to have ChatGPT. A well-known example is the use of the random forest model in the molecular neuropathology classifier in the pediatric domain. If you know that, it's become a very powerful, very accurate diagnosis tool that assists doctors to make diagnosis decision, which it helps to predict based on the DNA methylation data. In our research, we aim to build predictive models for CRISPR and drug response, as mentioned. Not only a powerful prediction model, but also we are able to find what contribute to the prediction. So they are the potential biomarkers. To achieve this, we are exploring both conventional machine learning method, such as random forest, elastic net, and deep learning approach like deep neural network, seeking the right balance between the power and the explainability. In our previous work, we found that predictive markers for a given gene dependency can vary substantially across adult or pediatric contexts. So what does it mean? A same gene vulnerability are actually explained by completely different underlying molecular network. So for example, the orange bars in the middle, if you can see there, these are the common ones. So they basically similar between adult and children. So an example is MDM2 dependency is associated with TP53 mutation in both scenarios. However, the genes highlighted in the purple bars, they are the ones we found they have different mechanisms, even though they all show vulnerability. So this is MCL1 dependency, I believe. Yeah. Then we can say the predictor for this gene is very different. So building on that work, we found is actually the DNA methylation landscape driven the MCL1 dependency specific to pediatric malignancy brain cancers. Specifically, is the BCL2L1 promoter methylation that regulates its own expression that marks whether the pediatric brain cancer will respond to MCL1 inhibitor. But it's different mechanisms in the adult high-grade gliomas. So this is really important, not only highlights we found a new biomarker, but it also shows that we need to look into pediatric cancer as its own. We can't really just repurpose whatever we found in the adult and then just translate it into children directly. We can't treat them as same cancer happen at an early age. It's biologically distinct. So DNA methylation here is a high quality and stable data set with also high translational potential, because if you know, we have an AIM-Brain project here. Now all the patients will be profiled for DNA methylation landscape. So whatever founding biomarker, if we found on the DNA methylation landscape, holds very high translational potential and then clinical readiness, translation readiness. So building on this, we refine our approach to focus on methylation sites that regulates gene expression and discover that several key dependencies in the high-grade glioma, including the succinate dehydrogenase on the right are the new data we haven't published, SDH complex involved in the mitochondrial energy metabolisms, and they are marked by distinct methylation patterns too. And if we look into using SHAP analysis, looking into the predictive model we built for the SDH complex, the CPG probes or sites are highlighted in the red box. Actually, they are in the same genomic cluster that regulates the calcium uptake in the mitochondria. So it's a very relevant function in the metabolic regulation in the energy production in the cells. So we're validating this finding, and hopefully we can find some new vulnerabilities and synthetic lethal interactions between these genes. We also move on to, deep learning models, as I mentioned, to further improve our predictive power. So using deep neural network, we are able to identify many more genetic vulnerabilities that can be accurately predicted with, more power. As shown in the chart and table, these are the outperformed prediction in using deep neural network compared to random forest. However, we are facing a problem that my PhD students are trying to tackle is the black box effect of deep neural network. We're trying to understand what make these models stronger. So we realize that it's normally a large combination of molecule signals collectively contribute to the prediction of this gene dependency. We can't really see any outstanding or enriched single features that can, mark a dependency. So that's something we're trying to extract key information to help design or do validation work. So the second project I'd like to share today focuses on identifying synthetic lethal interactions in the pediatric cancer. As I mentioned earlier, children's cancer often have very limited, strong actionable mutations, and single target therapies frequently fail. Therefore, combination therapies holds more promise, where identifying synthetic lethal interactions can open up many more opportunities. And the concept is very simple. So as you can see in the cartoon, two non-lethal perturbations, for example, gene A is a druggable target, gene B is commonly mutated, then they become lethal only when they were inhibited at the same time. So we're trying to find these interactions. Because we have CRISPR functional data sets together and we have a very genetic diverse collection of models, we are able to map out these genetic interactions and the functional consequences. So as shown in this workflow and the preliminary results, you can see that we, using the data set, we can very quickly narrow down from more than 5 billion possible interactions between any pair of genes down to below 200 pairs of these pairs that can lead to potential functional vulnerabilities or gained, outgrowth, tumor growth, then become a target for us. So you can see on the right top corner, again, TP53, MDM2, or MDM4, they are the positive controls as the tumor suppressor genes. On the right, we have identified several pairs of oncogene and their synthetic lethal partners. So they are the ones waiting for validation back in the wet lab. And then we hope to provide some hypothesis to these pairs too, and then see why they are specific to certain samples and then how they drive the synthetic lethal in those cancer contexts. These are the ones very readily to be used because we already know confirmed the mutation in these models, and then we have the other part are druggable genes, as I mentioned. But for a more experimental or discovery-based project, we're expanding this idea to look into epigenetic layer. So we want to combine epigenetic plasticity or adaptive ability and then see how we can combine this with other therapies. So we have added chromatin profiling in our CCMA pipeline. These are the markers we now profile on the selective 30 line, high-grade glioma lines. These are the active enhancer repressive markers and the promoter markers and also active gene body markers. And then using this and also a machine learning method called ChromeHMM, we are able to identify the chromatin status of each cancer subtype. So when we use the chromatin status as the core to align with other, to add on other omics modality on top of that to extract epigenetic regulatory network as the upstream, and then understanding what's downstream effect size of this status. Then we will feed back to the AI models to see is there any vulnerabilities is driven by epigenetic regulation. Then it also gives us insights. Maybe those vulnerabilities can be modulated, or it can be easily, adapted to gain resistant, or all these kind of responses. So now we only get 30 lines of data to play around. We firstly start with the gene level analysis and then we'll move on to more extended regulatory elements in the genome, such as enhancers or super enhancers analysis. And then we will add on to DNA methylation, expression, proteomics to understand as a whole. So firstly, we identified 10 chromatin states across these 30 lines, and then we group them because now we only have very limited data, very low resolution. We group these 10 states into 4 groups. So we have active states, inactive states, and also repressive states, and also, bivalent states. These are learned based on histomarkers we profiled, but to check whether we have gave right definition, we aligned it with the RNA expression level. Then it confirms we actually gives good assignment of these groups, because the active states showed higher expression levels generally than the inactive, than the repressive, than the bivalent. And if we look into the lines in closer, we can identify those genes. This is the active genes. This is the inactive and repressive genes. And then we identified those enriched active genes across different cancer subtypes in high-grade gliomas, which, a lot of them serve as positive controls. It should be there. So the ribosomals, the MHC, for T-cell response, ubiquitin, and neural development genes. And we have identified the signature genes for constantly repressive genes for each cancer type. Then these genes serve as the foundation that we can feed back to the CRISPR datasets to align which gene has an impact from the epigenetic or chromatin status impact for their functional readout. So to summarize, I know there's a lot of very endpoint results showing here, but I just would like to highlight all these data sets integrative strategies, because we have so many different omics modality. We can quickly find out the consequence of upstream, downstream effects of each changes were identified. So I hope I use my data set or results. You can see that multiomics and also AI frameworks enables the prediction of gene dependency and drug responses. And also you see more hope in the DNA methylation and epigenetic landscapes in joining the game, playing some role in identifying new therapeutic opportunities. And also synthetic lethal holds a lot of translational potential because we have a lot of model that identified potential pairs to just need validation in the lab, and then we can report back to the molecular tumor boards. Then experimental validation definitely is needed. We also want to focus on more epigenetic therapies and chromatin states to make more discoveries there. So this is the teamwork for sure. So the next generation precision medicine program is our group. So we have done a lot of wet lab work, dry lab work to generate this data, not only for us. We hope if you do work for children with cancer, please log on to the portal and download our data. Hope we can contribute to your research. And also our collaborators, national and internationally, and our funding bodies. Thank you. Yep. How can I answer questions? Okay, cool.
So we do have some questions from the chat.
Yeah.
And the first question is for DNA methylation CpG, have you checked the genomic proximity?
Generally, yes, definitely. But for the specific ones, we firstly start from the ones at promoter. We do have genomic location annotation. We group them as based on their, genomic functional, elements. So the ones I specifically talked is at promoter regions, but the one we published in the paper was at exon. Yeah, was that something?
Feel free
Example?
feel free to ask follow-up questions if that doesn't suffice, but we'll move on to the next question first. So what challenges do you face in applying multiomics to patient cohorts? So for example, sample sizes, tissue availabilities, and maybe costs?
As I mentioned, these are all cell lines because the beauty of that is we not only have molecular, we can do functional tests on them. So it can directly tie it with what, when we do something, we treat the cell lines, we know what will be changed. Then there is a gap. I have to admit, there is a gap if we want to directly translate what we see in the cell line in the tumor cohort. So what we do is based on the tumor data set as a pre-filter, we only look at the ones molecular features that agree between the cell line models and the tumor cohort. And then it gives us more confidence. Then we will be able to test it in the tumor tissues. And then the cost varies. And then I think it's more precise to get access to the tumors. And then if we can get is more than, happy to get something done. But often we need to, because they're really limited, we need to carefully choose what we can do, what is more interesting to do, because it's very small and then we can't do everything on those tumors. So cost-wise, haven't been, because most of the cases just we don't have enough tissue to spend money on. Yeah, at this stage.
So this is the last question. So what are the next steps or other technologies that you might employ in the future or in this project?
For for the pipeline work, as I mentioned, we are adding more proteomics and some PTMs in the post-translational modifications on the proteomics. That's definitely one direction we would like to do. And also we want to look into more rare cancers, for a lot of the children's cancer, they are N of 1, N of 2 in the field. And then we want to have more efforts there. But for research-wise, I probably want to go to more single cell level. We have crop-seq, which we do single cell level CRISPR and RNA sequencing, so we can understand it in a more granular level and then understand the heterogeneity in these tumors, hopefully. Yeah, cool.
Thank you very much.
Yeah.
Cool. Thank you. Thank you for that introduction, and thank you to Novogene for the invitation and the opportunity to tell you about some of the work that we've been doing. So this is just a project that we've been working on for maybe the last 2-3 years in the lab, and yeah, it's not completed, it's not finished yet, but it's we're hoping to get some good feedback on it. So any ideas or suggestions would be great. So the focus of the lab is really around, or the main cancer that we focus on is acute myeloid leukemia. And each year, is there the pointer? Yeah. Each year this is in Australia, around 1,000 new patients are diagnosed with AML, and it is essentially it is an immature. The leukemia is characterized by these immature cells that you can see here in this bone marrow or blood smear. These immature cells, which are called myeloblasts, and the presence of these cells in the bone marrow at a high percentage, so upwards of 20%, is what's required for diagnosis. It then presents as a medical emergency that requires treatment within days. And this last point here, treatment has not changed substantially in the last 3 to 4 decades. That's no longer true, but that was true when I started working on AML almost 10 years ago. And so it's amazing to think of how things have changed in this time. So yeah, treatment really didn't change for a very long time. You know, after the discovery of chemotherapy and bone marrow transplantation as probably the main treatments for a very long time. But really in the last 10 years, there's been an explosion of different targeted therapies to treat AML, and there are various different targets that I won't go into in too much detail, but just to mention that there are, you know, these probably summarize the main drugs that we currently have available against AML. Aside from chemo, many of these have been approved by the FDA only in the last 2 or 3 years. And it's important to have this arsenal of drugs to treat this very aggressive leukemia, because single agent efficacy is most often not durable. So patients will have a response and then they will relapse. And so the main question really for not just clinicians in the AML space, but just across the board for all cancers, is how to combine them effectively. Similar to what you heard from Claire. And so this is, you know, something that scientists and clinicians think about a lot, and it's a pretty simple idea, right? Combination therapy is you're combining more than one drug, you know, 2 or more drugs together, in order to try and achieve some kind of synergy between the 2 drugs. And that often comes at the expense of toxicity, right? You may have increased toxicity as a result of combining 2 different drugs together. Now, the idea of sequential therapy is that you give one drug after the other, rather than combining them together. And the idea there is that although your efficacy is more likely to be additive when you combine when you give the drugs one after the other, the idea there is just to minimize the side effects and reduce the toxicity. And so that, you know, although you get less toxicity, it's at the expense of the increased efficacy that you might see. And this idea, this type of sequential therapy is often employed in the clinic out of necessity, because combining the drugs is just too toxic for the patients, and the side effects are unbearable. So they may, out of necessity, do that. And so we started thinking about, well, what's a rational sequential therapy approach? A rational sequential therapy approach might be to give drug A, and this drug would have to reprogram the cancer cells. It would have to change their state so that they're now more sensitive or more vulnerable to the second drug B. And hypothetically, the idea here is that by giving the drugs in sequence and having one reprogram the cells before the second drug, is that you may maintain that synergistic efficacy that you see with the combination treatment, but without that overwhelming toxicity that you might expect, because you're giving the drugs separately. Of course, this is hypothetical, but the concept in theory should hold true. And so, you know, what's essential for this idea of a rational sequential therapy is that you're inducing a cell state transition prior to the second therapy, so that you're priming the cells to become sensitized to the second line treatment. And yeah, and like I said, it's a potential to maintain that level of efficacy while sparing that toxicity. And so when we say cell state change or reprogramming of the cells, what do we mean by that? Well, all cells have to maintain their identity, and they do this through something called epigenetic memory, in part. And epigenetic memory really just refers to the ability of all cells in our bodies and across life, is the ability of cells to remember gene expression patterns. They have this ability to remember, like a colorectal, a colon cell remembers that it's a colon cell when it divides, and although that seems quite obvious, it's actually a complicated process that we're starting to understand in more detail. But the idea is that gene expression programs persist through cell division, and that they're remembered, and whatever that memory is, it's independent of the initial input. And the way the field currently thinks about it is that there are two main or broad processes or ways by which cells remember what they are. And the first one is this idea called a gene regulatory network, which is like GRN memory. So it's like network memory where every gene is connected to all the other genes, and there's kind of like a massive feedback loop or many feedback loops that are interconnected, and these genes regulate one another. And that memory depends on network interactions, like gene A regulating gene B and gene B regulating gene C, etc. Epigenetic memory, on the other hand, it can be thought of also as feedback loops, but at the locus, at individual genes. So individual genes can have modifications to the chromatin state, which can keep that chromatin, keep that gene in an off state in order to generate this stable gene expression state. And so this is independent of the network. It's independent of continuous inputs. It's kind of like, you can think of it as like local memory. So each gene individually can have this local memory or epigenetic memory, whereas the GRN memory is more like a global network type memory. And so, one of the questions we had was, well, is exposure to targeted therapies in cancer, is that remembered by cancer cells? What capacity do cancer cells have to remember different perturbations from different therapies? And so, thinking back to leukemia and hematopoiesis, is that when you have, this is normal blood development where you have a stem cell, hematopoietic stem cell, that differentiates down to the various effector cells of the immune system. And in this process, there is extensive reprogramming of the chromatin state and the networks. So there's a constant transition from one cell state to another. And obviously that requires a lot of memory and changes in gene expression, as you might expect. When leukemia happens, when leukemia is formed, you get this block in differentiation. Cells accumulate. They're unable to differentiate further, and they lead to disease. And there are many epigenetic regulators that are involved in mediating the chromatin state and the memory of these different cell types that happen during development and during hematopoiesis. One of these are some of the most important ones, or some of the very important epigenetic regulators that are required to mediate that chromatin memory that I was referring to. And the one that's involved in activation is the compass complex, or MLL1 and 2, which methylate lysine 4 on histone 3. And then you also have the repressive complexes, in this case PRC2, which methylates lysine 27 on histone 3. And these are involved in repression and memory of the silencing that they mediate. And this regulatory, these epigenetic complexes are frequently dysregulated in leukemia as well as many other cancers. And so the idea is that this important axis of regulation of epigenetic memory is frequently co-opted in different cancers, and particularly in acute myeloid leukemia. And so one of the mutations that is fairly common, and leads to an aggressive leukemia is this mutation in the MLL1 gene. And this mutation is basically a chromosomal translocation, which cuts the MLL protein around here and fuses it to these different fusion partners, which dysregulates its function. So MLL1 is now truncated, it's fused to a different partner, and it functions differently. This MLL fusion protein is a very potent oncogene that causes very aggressive leukemias in children and in adults as well. And there are various mechanisms by which it regulates gene expression, which I won't go into too much detail, except to say there are various epigenetic proteins or enzymes that are recruited to chromatin to modify chromatin. But what's important to mention is that recently these so-called menin inhibitors have been developed to treat AML, this type of AML in particular. And these menin inhibitors block the interaction between menin and the MLL fusion oncogene, as well as the wild type MLL1 protein. And they basically displace the protein from chromatin, and they shut off gene expression, these menin inhibitors. And they've been very effective at treating these cancers, these leukemias, and the FDA approved menin inhibitors for relapsed refractory AML last year, I think. And they're currently in ongoing clinical trials, and it's a very exciting time for people working with menin inhibitors and clinicians treating patients with these types of mutations. As I mentioned, there's lots of clinical trials now happening that are combining menin inhibitors with different therapies for leukemia. And so what we first set out to do was understand, okay, what happens if we give the menin inhibitor, if we treat cells with the menin inhibitor for a long time, what effect does that have on the cells? How does that affect epigenetic memory? And so to do this, we developed this in vitro model of adaptive resistance to menin inhibitor. So we took some leukemia cells, we sorted single cells and expanded those single cells into large populations of cells, and that's just to minimize any of the genetic heterogeneity that you often get with cells that are just grown in the lab for a long time. And so we've got these two populations. We then treat them with the menin inhibitor for increasing concentrations of the drug over many months, so around 3-4 months, until we get a population of cells that are resistant to the menin inhibitor. And so what we found was that following the development of this resistant state, following long-term exposure to the menin inhibitor, we're able to culture these cells in the absence of the menin inhibitor. And this is just to show the validation to show that these cells are in fact resistant. So these are the drug-naive cells, and you can see they're very sensitive to two different drugs. This is the menin inhibitor and also a DOT1L inhibitor. You can see that the cells really stop growing and they die when you treat the naive cells. But in the resistant populations, you can see that they are able to proliferate in the presence of the menin inhibitor. And what we found that these resistant cells, these resistant populations, really showed no mutations in menin, MLL1, or any recurrent mutations that might explain the resistant phenotype. When we performed RNA sequencing analysis, we found, quite interestingly, that many of the target genes of the drug, so this is an example of those, MEIS1, this is one of the canonical target genes for this menin inhibitor. And the reason people think the patients respond really well to menin inhibitors is because MEIS1 is this critical target gene. And you can see here that with the drug in green, expression goes right down. This is in the naive cells. But then in the resistant cells, it's already down. So MEIS1 is already silent in the resistant population. And so we see this even in the absence of the drug. So this is the resistant cells cultured in DMSO. And this tells us that there's stable silencing of MEIS1, which may be due to epigenetic memory, or it may be due to just reprogramming of the cells and a completely different cell state. Because remember, we cultured these cells over 3-4 months in the drug. And so this brought up some questions for us, does menin inhibition induce epigenetic memory? Is that why we see stable silencing of some target genes? And can this memory induced by therapy be harnessed? Can we leverage that memory for therapeutic benefit? And so what, Grace and Daniel did in the lab was that they did some simple experiments where they treated the cells for a very short period of time. So 7 days rather than, 3 months. Treat the leukemia cells for 7 days and then wash out the drug. So then there's no drug for 7 days. And then we did RNA sequencing. And what surprisingly we found was when we washed out the drug, a lot of the transcriptional changes were maintained even after 7 days without the drug, compared to this is just on drug. And so if we look at this heat map, you can see that some of the changes do in fact stay, are sustained or persist in the washout sample, which you can see here. But some of the changes do rebound back to the baseline, back to the normal state of the cells. This is just showing an example in a different leukemia cell line, where we see very similar patterns, where we see many genes that stay downregulated, as you can see here, following drug withdrawal. And so this suggests that the cells really do carry a memory of the exposure to the menin inhibitor. If we then do some clustered analysis, we can see that there are genes that recover when you wash out the menin inhibitor. There are genes that partially recover, and then there are genes that seem to be stably repressed after we wash out the drug. And so this suggests that the degree of recovery does vary across genes downregulated. We then did some long-term washout experiments. We went up to 63 days and we collected samples at various different time points, and again performed RNA-seq in 2 different cell lines over many different time points. And you can see just this one example here for MEIS1. In one cell line, it kind of slowly, gradually returns back to baseline expression. And then in one cell line, it really persists. The silencing persists for up to 63 days. And so this heat map, again, doing some clustered analysis, showing that there are genes that rapidly recover over time. This is up to, like as I mentioned, 63 days. And then there are genes, really interesting group of genes that gradually change their expression. And remember, these cells are dividing. They're perfectly fine and dividing over 63 days, gradually recover their expression. And then there are genes that show this long-term repression. So we recently did some single-cell RNA sequencing analysis to understand, well, what's happening, at the single-cell level, not just at the population average. And so this is the DMSO here in dark blue. Light blue is the menin inhibitor treatment. And then all the washout samples are kind of clustered together over here. And if you look at the time points, you can see that they kind of start out in yellow here. This is 6 days post-washout. And then you've got 12 days over here. And then 18 days. Oops, I think the colors are all mixed up. Oops. But trust me when I tell you you can see that the cells under treatment are here, and then they gradually revert back to the original state, but they never quite make it back, at least not at this time point, which is 18 days, and that's in red. And if we look at some genes, the expression of some genes in these samples, we can see, for example, MEIS1, the one I showed you before, you can see it's completely down in the treatment, and then it kind of gradually returns back to normal expression in some cells within the sample. And so what we think is going on here is that and what's quite clear also is just from the how wide the distribution of the cells are when we wash out the drug. It's almost like they become much more heterogeneous and the cells become destabilized to some extent. And so this is something that we're looking into further to gain a better understanding. This is just a proliferation assay to show that the cells treated with a menin inhibitor, VTP in red, they at the point of washout, you can see that the cells don't quite, they whilst they proliferate after drug withdrawal, you can see that the proliferation rate is lower than the vehicle-treated cells. But the most important thing here is also to show that other drugs So other AML therapies, for example, a FLT3 inhibitor or IBET, whilst they have a massive effect on the cell proliferation and cell survival, when we wash out those drugs, the cells completely restore their normal proliferation rate. So you can wash out these toxic targeted therapies and the cells, a few days later, the cells don't care. So there's no anti-proliferative effects that persist following drug withdrawal of these two drugs. And so we thought, like the cells are carrying this memory of the menin inhibitor, and we made them resistant now, because previously I showed you that, long-term exposure to the menin inhibitor and you get resistant cells. Now we've got very short treatment. Is that enough to maybe induce resistance? So what we found was, we did the first treatment, we washed out the drug, and then we put on the second treatment. If we just put the menin inhibitor back on the cells, we actually find that the cells are more sensitive. So we get a more induction of cell death if we put the menin inhibitor back on cells that were already, treated with the menin inhibitor previously, as you can see here in this last bar graph. And in fact, the washout cells treated with the menin inhibitor again have increased differentiation, or they differentiate faster. They're almost primed to differentiate. And so this made us think that maybe we could harness this reprogramming that we're seeing, and that maybe this epigenetic priming can be used for sequential therapy. So the idea is that, we treat with a menin inhibitor, wash out the drug, and we've got these transcriptionally rewired tumor cells. Can these have new vulnerabilities now? That was kind of the question that we were asking. And can we harness this? And so we did the same experiment. We treated with the first treatment, washed out for 7 days, and then put on the second drug. And if we do that for a variety of other therapies for AML, such as venetoclax or IBET-151 or the FLT3 inhibitor, gilteritinib, you can see that in all of these cases, the cells show this increased sensitivity to these drugs. So here is an example of one. So this is menin inhibitor first, and then DMSO. So you can see that the cells, there's maybe a little bit of background cell death here. But if we just put on venetoclax on its own, after DMSO, just on normal cells, you can see that there's some induction of apoptosis. But if we put venetoclax, which is an AML therapy, if we put that on after the menin inhibitor, after priming with the menin inhibitor, you see that there's a much bigger induction of apoptosis. These are just some IC50 assays to demonstrate the shift or the change in sensitivity for these two cell lines. And this is quite a nice experiment because here we've flipped the order. So if we give venetoclax first and then the menin inhibitor, you can see that the amount of apoptosis is far lower than if we give menin inhibitor first. And so the order of the treatment really matters in sequential therapy. So it matters that the first drug is inducing some kind of reprogramming or rewiring of the cells, which makes them more sensitive to the second drug. And so just, we wanted to compare the sequential therapy treatment to combination. What happens if we just combine the drugs? And so we did an experiment where we combined them in the first half of the experiment. So the first 3 or 6 days, we combined them, or in the second half, and then compared it to just giving the drugs one after the other. And what the takeaway message here is that giving menin first and then venetoclax in purple was better than both combinations, either giving the combo first and washing out the drugs, or giving the combo in the second half of the experiment after DMSO. But it wasn't better than giving the combination throughout the duration of the whole experiment, which is in this brown color. And the sequential therapy induces, as I showed you before, induces a lot more apoptosis than even the combination for the same amount of time. And then, the effect on cell number is comparable between the combo and the sequential when we look at that. So just lastly, I want to just show you how we're looking at the effects of different drugs on memory, on how cells remember the perturbation. So if we compare these different targeted therapies for chromatin factors that are involved in epigenetic memory, such as EZH2, DNMT1, as well as the menin inhibitor, of course, compared to drugs that don't target chromatin factors involved in memory, but have big effects on network memory. They disrupt gene expression programs. So we did very similar experiment. We treated with these drugs, washed them out, and then cultured the cells for a period of time, and then sampled the cells to look at the transcriptome. And you can see here all the different gene expression changes that we get with these drugs. And then in the washout samples, you can see that with IBET-151 and gilteritinib, which disrupt the network, you can see that all of the changes revert back. Pretty much all of the changes come back, and there's not much remembered by the cells. But if we treat with EZH2 inhibitor or DNMT1 inhibitor, which is the affects DNA methylation, of course, you can see that a lot of these changes persist in the washout samples. And so here's just an example of some genes. IGF-1, you can see that this gene is induced by menin inhibitor somewhat. It's induced a lot by gilteritinib, the FLT3 inhibitor. But in the washout samples, you can see that expression of IGF-1 goes back to zero, just like in the parental cells or in the vehicle-treated cells. But with the menin inhibitor, you can see that there's some memory of induction of this gene. MYC, which is a target of the BET proteins that are affected by IBET-151, you can see expression goes down, but in the washout, it jumps right back up. And this is another gene where there's memory following washout of the menin inhibitor. You can see it goes down and just stays down. This gene is not coming back up. And so this is saying the potent perturbation of gene regulatory networks isn't sufficient to induce a memory, at least with the perturbations that we've used. However, direct disruption of chromatin-based memory mechanisms can and does lead to persistent expression changes. And so the way we're thinking about it, this is the second last slide, is that you've got cells that are sitting in the Waddington landscape. If you apply a signal or a pressure or a perturbation, the cells will kind of move, maybe perhaps up the hill, with a strong external signal. But if you remove that signal, if you remove that perturbation, the drug or whatever it may be, the cells can just kind of go right back to their original state. And that's obviously a very important mechanism that protects cells from changing too easily. But if you co-opt chromatin memory as well, not just network memory, if you disrupt chromatin memory or co-opt it, you might find that when you push the cells to this other state and you change the landscape by disrupting those chromatin mechanisms, you can alter you can stably alter cell state from which it can't return. And so we're pretty excited by some of these insights that I showed you around sequential therapy, and that we've got a collaboration with the drug company that manufactures the menin inhibitor, as well as clinicians at the Alfred Hospital, to hopefully do some clinical trials around the idea of sequential therapy, because it's important to, as I mentioned at the start, with combination therapy, that we're not going to be able to combine more than 2 drugs very easily, right? There will just be too many side effects and too much toxicity associated. As soon as you start to combine 3 drugs, 4 drugs, that's never going to be possible for cancer. And so we're going to have to think about ways to add drugs in sequence. If there's 4 drugs available to a patient, you can't combine all of them together. But if you know which drugs should be given first, because they may reprogram the cells, or they may prime the cells to respond better to the following drugs, that's really important information to clinicians. And so we're hoping to find contexts where we can really confirm the efficacy of sequential therapy. And so that just remains to thank the people that have done the work, all the people in the lab, especially Daniel and Grace, who have kind of driven this project, as well as other labs that we've collaborated with over the last few years. Yeah, thank you.
Thank you, Omer. I feel like your last slide had just answered this question, but I'm still gonna read it out anyways.
Yeah.
So suppose menin inhibitor is the drug A. Are there any possible drug B candidates for additive treatments?
Yes. So venetoclax, FLT3 inhibitor, these are, you know, both current AML therapies in the clinic, and we find that the menin inhibitor primes leukemia cells to be much more sensitive to both venetoclax and the FLT3 inhibitor. So those would be two good candidates for doing some, you know.
Yeah.
Yeah. Is that the question, or was
Yes.
it additive in? Yeah.
Yes, that is the question. Thank you. And the second question is, are there any other diseases that are currently treated using the concept of epigenetic memory, or is this a novel concept for leukemia?
So I haven't seen people that have explicitly mentioned epigenetic memory as the mechanism for what's happening, but people have used epigenetic therapies to try and prime cells to respond more to immunotherapy, for example, right? People have done stuff like that, but it's generally not through epigenetic memory. It's usually through just inducing the expression of some immune protein and then hitting the cells with that immune drug, immune therapy, to kind of exploit the change.
Okay.
You know, so it's usually that type of thing. So I think it's fairly, you know, hopefully.
So it's a more
A novel-
specific mechanism.
Yes.
Yeah.
Yes. Yeah. And it's more durable and persists for longer than, yeah.
Yeah, that's very cool. We have one more question from Evelyn. So what are the major challenges of applying these findings to a clinical setting?
Yeah, that's a good question. So I think drug companies don't like to stop the treatment. and this is basically telling them to stop the treatment, right?
Yeah.
Because it's a sequential therapy thing. They want to just give the drug as much as possible. Continuously and in combination.
Yeah.
So effectively we're saying, no, you give it for 2 rounds of treatment and then you stop and then you follow it with a second drug. You know, which and the clinical setting, from talking to clinicians, the clinical setting that's probably the most promising is in the elderly patients who can't tolerate combination. Like they can't tolerate multiple drugs at the same time. So that would be a good place to test it and see whether it's, you know, it's tolerated and it still has good efficacy.
So a good angle is the elderly patients and then start from there.
That's right for now. But I think the concept should apply to all patients, because although combination does work and is effective, and I'm not saying don't do combination treatments, I'm just saying that if you've got 2 drugs that work in combination, maybe you can bring in a third drug, for example, and prime the cells, prime the cancer or the leukemia to respond to the combination. Because you're never going to be able to do triple combinations are very rare and problematic, and often they have to reduce the doses, right? There's often dose limiting toxicities. And so, yeah, that's kind of the idea.
Thank you very much, Omer.
Oh.
You have a question?
Yeah, actually, I want to ask, do you know what changes after you prime the cells, like post the memory? You know what I mean? Like,
Chromatin changes.
yeah.
Yeah.
What's the epigenetic
Mechanism. Yeah, we've got lots of data on that. I just didn't put it in because I didn't want to like complicate it. It's K27 trimethylation mainly. And we think some DNA methylation as well. Yeah.
Thank you, Omer, for the wonderful talk and the insights. Thank you, Claire, for the question as well. Because of time's constraint, I think we'll stop here, but feel free to reach out to us or the speakers for any follow-up question. Thank you all for coming here today.